Do you know who is this? Spider-Man. Okay, Spider-Man has a super healing power. So you know that he is fighting to protect humanity. And sometimes when he injured, he can immediately heal. Not too fast, but at least he takes about 2 days to get complete healing. However, his super healing power is not as great as the Wolverine on the Deadpool. So the dream of humanity is as old as, the dream of immortality is as old as humanity. So I would like to bring you to some stories. You will realize that my whole talk, I will relate to some Greek mythologies and the history of humanity. So in a Greek mythology, Achilles has been foretold. The mother of Achilles, Titus, has been foretold that Achilles would die at a very young age. So in order to prevent death, the mother has brought Achilles to river Styx, and she dipped Achilles into the river Styx. So she hold Achilles on the feet here at the heel, and he dipped him into the river Styx, which is supposed to grant him the power of invulnerability and immortality. So what's next is Achilles grown up to be a hero, and he has won many wars, and the most notable feat was in Trojan War, where he managed to slay off Prince Hector in the Troy Gate. However, at the end of the war, Achilles was somehow being shot by Paris. So he was being shot at the heels. And unfortunately, he has been killed just by that poisonous arrow. The reason why he was killed is probably, according to the Greek mythologists, is that the heel was not deep into the river Styx, and therefore this part of the body is not being protected, and therefore he cannot heal from the injury. So the name Achilles tendon, which connect the bone on the heel and also the muscle, is named after Achilles, and it is called as Achilles tendon. Can you see? Is it very dark? So now I want to fast forward the history a little bit to arrive at the ancient Egyptian. So the ancient Egyptian, as you know, they mummified their bodies, and they believed that death is only an interruption in their life. In fact, it is not a cessation of life. So they bring together with them their loved items. For example, perhaps their animals, their clothing, whatever, that they think that can accompany them to their eternal life. So now, again, I fast forward the history again. So humans tried many ways to salvage our life. So we try many ways to awake even the dead. So Luigi Galvani, in the very early in the history, has found out that if we apply some electrostimulation onto the body, the muscles can contract. So therefore, he has published many papers about animal electricity. I'm so sorry about the slides. I have to turn on and turn off. So after his death, his nephew tried to prove whether his theory worked or it doesn't work. So his nephew went to do a public demonstration. So he used a corpse. So this corpse is an executed criminal which has been hung, and he's confirmed that at that time. So what he did is that he applied some batteries onto the bodies, and after some time, the jaws of the criminal has quivered, and the muscles on the face start to get distorted. And suddenly the hands are raised up and clenched, and the legs start to move, like dancing. So this shocked everyone. It's very spooky to the public. So of course, the executor who tried to execute him again have to re-execute him because it is not confirmed whether it is dead. So it is quite spooky at that time. So the science and the technology has tried to find or discover many reasons or the mechanisms that can cause aging. There are 3 theories related to aging. For example, the first one is the oxidative damage theory. So we understand that the deleterious effects of the reactive oxygen species can cause the damage to many cellular constituents, and it involves the molecule of oxygen and the oxidative enzymes. So we also understand that when DNA is being damaged, for example, by radiation, so DNA can be spliced, and if our body doesn't have sufficient enzymes to repair the DNA, so the DNA in the cells can get mutations, and therefore it can dysregulate the gene expression in the cells. And therefore, because of this dysregulation, it can cause us to age because the cells died. So then there is another epigenetic theory. So in this theory, it says that there could be some hypermethylation in the CpG island in the cells. So the more is the hypermethylation, the more aging is the cells itself. So therefore, this theory also proposed that we can actually identify the biological age of someone by looking at the pattern or the degree of hypermethylation. So of course, aging is not a disease, but aging is associated with many diseases like cancer, neurodegeneration, frailty, aesthetic, the wrinkle, heart disease, arthritis, type 2 diabetes, and stroke. And these are just some of the lists. So again, there are many researches that has been done to study what are the ways that we can prevent or promote longevity. Prevent death and promote longevity. So worldwide, there are 5 regions in the world, and the top 2 ranks are that have the highest concentrations of centenarians are in Okinawa in Japan and also in Sardinia in Italy. So here what I'm showing is a group of pop sensation, which comprise of 33 grannies, and the age of these grannies, the average age of these grannies is 84. So they have been named as a KBG 84. So they are so active, in their lifestyle. And in fact, in 2015, they have released a single called as Come On and Dance, Kohama Island. So this is a group of male centenarians socializing. So there are many researches that have been done to look at what are the biological, physical, mental, and the lifestyle activity that can help them to age very healthily. Of course, healthy aging is very important because it actually helps to reduce the burden in the healthcare system and also to help to promote the economy in a country. So in the modern world, there are many products that have been produced in order to slow down aging, maintain youth, or to prevent diseases. For example, these are the lists that you can get over the counter, probably from the pharmacies. So there are products like vitamins, antioxidants, perhaps some injections, probiotics to help to maintain the gut health. So despite that, there are all these products that we can find in the pharmacies. The efficiency of these products to slow down aging is still not well proven. However, the scientists keep on exploring to find ways to find treatment or any alternative methods to reduce or to slow down the aging processes. So the very advanced type of technology that has been established includes cell therapy and gene therapy. And last year I've spoken about gene therapy, so I'm not going to talk too much on gene therapy. In fact, I will just touch on the cell therapy. Cell therapy means we can use cells to replace the dying cells in our body. So it is true. Okay, because we understand that cells basically they can divide in a binary manner. That means one cells will divide into 2, 2 divide into 4, 4 into 8, 8 into 16, 16 to 32, and so on. So this is quite true, and this theory of cell division that all cells come from cells has been proposed by a very famous German pathologist named as Rudolf Virchow. So in the Latin, the epigram was omnicellulae e cellular. So it means that all cells come from cells. So to illustrate further to you and to make you to understand a little bit more, so we understand in our blood circulatory systems, okay, we have the white blood cells, red blood cells, and the platelets. And we know that physiologically, the red blood cells, they die after 3 to 4 months. And all these cells can be renewed. And if you trace back the lineages of these cells, for example, the white blood cells, the red blood cells, and also the platelets, if you trace back, you will find that they are actually derived from 2 progenitors, which is the lymphoid progenitor cells and also the myeloid progenitor cells. And if you trace further again, you will find that these 2 progenitors comes from the multipotential stem cells. And all the multipotential cells comes from hematopoietic stem cells, which actually congregate in the bone marrow. So the hematopoietic stem cells give rise to all kinds of blood cells in our body. And in fact, hematopoietic stem cells is one of the champions in the stem cells because they can actually proliferate in the culture, that means in the lab. If you isolate from an individual, you culture them in the lab, they can be cultured as long as the 5 generations of a living mice. So now again, I want to bring you back to another fiction whom many of us might have read or even watched. This is about the Dracula. So there is, I want to bring this because I find that this is very interesting. So it tells you about the blood being a very potent symbol of life. And in this fiction, it also tells us about the science behind blood transfusion. So if you remember about the Dracula, so they survived, they are vampire, and they survive by biting on their victims to suck the blood. So they survive, they suck the blood in order for them to survive. And the first English victim of Dracula is Lucy Van Westenra. So in the night, at night, Dracula would suck the blood from Lucy Van Westenra. So Lucy Westenra becomes very ill, and no one actually knows the reason why her health is declining until one day Dr. Seward asked Professor Van Helsing to come over to check on her. So Professor Van Helsing had to do the blood transfusion to her in the morning. So in the morning, she received a blood transfusion, and at night, she was being sucked by Dracula. So somehow, she managed to live for some time. So I want you to see that blood can be transplanted into another individual, and they can still repopulate in the recipient. So by the way, Dracula is authored by author Bram Stoker in 1897. So that was a very long time. And at that time, they already have an idea about blood transfusion. So just now I mentioned about the stem cells. Okay, so what is actually stem cells? So stem cells are cells that are capable of making many of them so they can replicate so that our body will not be lacking of these cells. So that because we know that they are very important to replace the dying cells. So other than that, they can also become specialized functional cells. So here, the process in which they become specialized functional cells is called as cellular differentiation. So basically, a stem cells, they can divide in 2 manners. The first one is a symmetrical division, and the second one is through asymmetrical division. So the symmetrical division will give rise to 2 daughter stem cells, which have the same properties of the mother cells. So this is important to maintain the reservoir in the tissue, so that if there is any injury to that specific tissue, these stem cells can continue to divide and supply endless amount of cells that helps in the function of the tissue. So in an asymmetrical division, one stem cells can divide into a stem cell and 1 differentiated cells. So the differentiated cells that I mean here are those matured cells that forms our body tissues. In our body, we have got about 200 types of tissues in the organs. So example Excuse me. Yeah. So there are about 200 over cell types in our body. For example, there are sex cells, immune cells, epithelial cells, nervous cells, blood cells, bone cells, muscle cells, and the fat cells. So the stem cells is present in our body for replacement of damaged cells. So now, you may ask me the questions, where can we find the stem cells? And I'm sure some of you might have heard about embryonic stem cells. So the stem cells, the first stem cells that is derived during human development happens when the ovum is being fertilized by the sperm. So the moment they are being fertilized, this ovum will become zygote. They will start to divide again in a binary manner. So the zygote will divide into many cells, which then they arrange themselves into a ball of cells known as the morula. And then they become blastocysts, and then they started to become, to really assemble themselves to become the embryo and then develop to be fetus. So here, the zygote would have the highest potency as stem cells that can give rise to all many kind of cells, because from the zygote can differentiate into morula, which can form a total embryo or a human being. So from here, once they become the morula, they will arrange themselves to become the blastocysts, and the cells inside here, which is known as the inner cell mass, is called as the embryonic stem cells. So that means these embryonic stem cells can give rise to all the type of cells in our body. So as these cells rearrange themselves, in the tissue patterning or self-organizations, so some of these cells will be asked to go to form the head, to form the limb, to form the heart, to form the lung. So when they are traveling along the embryo plane, some of the cells, the stem cells, are being stuck in that specific tissues. And therefore, that is the reason why is that we can find certain amount of stem cells in our adult life. So therefore, we can harvest the stem cells from the baby or even an adult or from the elderly. However, I have to mention that the quality of the stem cells would differ. So the quality of the stem cells from younger person would definitely be better compared to an elderly person, because they have not been exposed to any agent that can damage the DNA. So I have to mention here that at this site, the proliferation and the differentiation ability of the stem cells is higher compared to stem cells that you can harvest in the adult life, because adult cells here are more mature compared to the stem cells at this site. So basically, the function of the stem cells during embryonic development is very helpful for organ development, whereas on the other side here, it is much more useful for repairing damaged tissues. So again, here I want to show you about the embryonic stem cells. So when the ovum is being released from the ovary, so the ovum will normally be fertilized in the fallopian tube, and they will start to divide, and they travel along the fallopian tube to reach to the uterus. So for human, you can harvest the embryonic stem cells on day 5 to 7. So there is a certain time that you can harvest the embryonic stem cells. So you cannot harvest if the embryo has been implanted for too long, because these cells will lose its pluripotency. So then you may ask me, okay, where else in adults can we harvest the stem cells? So we can harvest the adult stem cells from different regions in our body, including the tooth. So in the tooth, in the dermal papilla, we can isolate the mesenchymal type of that stem cells. Other than that, we can also isolate from the hair follicles. So, in the hair follicle, we can get epithelial stem cells or the mesenchymal stem cells. And that explains why is our hair, they are able to grow longer and longer, even though we have cut them. So other than that, we also can isolate the stem cells from the liposuction tissues. So the liposuction tissues is normally done in the abdominal area. So when we isolate this kind of stem cells, normally these stem cells is responsible to generate the fat cells. So other than all those tissues that I have mentioned, actually the list is quite many, but I just highlighted some of them. So other than that list, we can also isolate the stem cells from the human umbilical cord. So from the human umbilical cord, we can, from the Waters jelly, we can mince those tissues and then culture them into on a petri dish. So when we culture, we'll be able to find these adherent cells with fibroblastic look. So these cells can be expanded and it can be differentiated. It can be differentiated into osteocytes, chondrocytes, or the bone cells. So compared to embryonic stem cells, the isolation of embryonic stem cells, the isolation of mesenchymal stem cells from the Waters jelly is much more advantageous. The reason is because the umbilical cord is considered as a medical waste. So therefore, you do not need to kill an embryo in order for you to harvest the embryonic stem cells. Therefore, it causes less ethical and religion controversy. So now, I also would like to touch about another type of pluripotent stem cells that behave like the embryonic stem cells. What if I tell you that we can actually get cells from our body now and reprogram them to be embryonic stem cells? Would you be excited to know about it? Right? So Professor Shinya Yamanaka has done research to look at what are the genes that can reprogram the somatic cells to become embryonic-like stem cells. And he found that there are 4 important genes. That's the OCT4, SOX2, c-MYC, and the CLIC4. So when he inserted these proteins into a fibroblast cells from the skin in a culture, when it is injected into all these cells were then reprogrammed to acquire the pluripotency similar to the embryonic stem cells. And then these embryonic stem cells can be used to make the different kind of cells, the 200 type of cells in our body. For example, the fat cells, the blood cells, neural cells, skin cells, and pancreatic cells. So this type of reprogrammed cells is called as induced pluripotent stem cells. So now you may ask me, what is the significance? Why do we want to turn these cells into embryonic stem cells? The reason is it would be very nice if we can prepare cells that we want to repair in the future. For example, if I expect myself to have a knee problem in the future, perhaps I can use the cells from my body, I can turn it into induced pluripotent stem cells, that I can then induce these cells to become cartilage and transplant into my body later on in the future. So that is the reason why everybody is rushing, to find ways to make the pluripotent stem cells. So Professor Shinya Yamanaka won the Nobel Prize laureate in 2012. So I have mentioned to you about cellular therapy in the earlier slides. So we wanted to harvest all these kind of stem cells because we want to harness the power of these stem cells and to make or to induce stem cells to become different type of cells. The purpose is to replace the dying cells that happens in many diseases type. For example, in the Alzheimer's disease, there's an imbalance of the protein expression, for example, the tau protein. presence of very high amount of tau protein can cause the neuron cells to die. So if we can induce the stem cells to become the nerve cells and we inject the nerve cells into the patient, so probably we can find an alternative strategy to treat Alzheimer's disease. So other than that, we can also induce islet cells, cardiomyocytes, hepatocytes, glial cells, chondrocytes, and nerve cells to treat, if not to stop these diseases. So how do we make the stem cells to become the functional cells that we want? So we can harvest these stem cells and then treat the stem cells with different chemical agents. For example, in my own research, I have been trying to turn the stem cells to become the retinal neuron cells. The retinal neurons is very important for visual functions. So what I did was I have to take the stem cells and then I incubate with the retinol acid for a certain period of time. And then after that, I will be able to observe that there is changes in the morphological. And then I also study on the functional capability of these cells to confirm that these are truly the retinal neurons. So from here, this is called as the undifferentiated stem cells. And the cells that we make is called as the differentiated cells. That means the stem cells are being induced to differentiate into the matured specialized cells. So there are also many protocols that have been established. I'm just giving some examples. So this is a method to make dopaminergic neuron differentiation for Parkinson's disease. So the protocol is established based on the starting material of the stem cells, because you know that we can isolate the stem cells from many different tissues in different stage of life. So here, this protocol is suggesting to use the stem cells, the embryonic stem cells, and then we have to induce or treat the cells to form the embryonic body. So this embryonic body is actually mimicking the blastocyst during embryonic development. So to make the embryonic body, we have to add in B27 and N2. Then further, after some time, we have to add SMI and FGF2 to make the cells to become neural rosette. Then after that, to further induce them to become neurogenic lineages, we have to add different kind of chemicals. And to monitor whether they have truly formed the neurons, we have to look under the microscope and also to study the markers and also the electrophysiological function of the cells. So how about to make the heart cells? So again, here, this diagram shows the method to make the heart cells from the pluripotent stem cells. So you may make the pluripotent cells to form the heart cells by culturing them in suspension method or making them to adhere onto the petri dish. So it is up to you to decide on the methods. And then after that, you can observe whether the cells can function. And now I'm going to show you a very short movie about the beating of the heart cells. So this is taken from the internet, from the company Mutiny Biotech. So in Malaysia, we also have researchers who have successfully reported of making the heart cells that can beat. So now I want to bring your attention to the current clinical trial state of stem cells. So we have tried to harvest the stem cells and we want to induce them into different kind of cells. So in the clinical trial, there are phase 1 and phase 2 that has been reported to studies on the safety and also the efficiency profile of the stem cells. So the clinical trials that have been studied use the embryonic stem cells, okay, to make the oligodendrocyte progenitor cells, which is the neuron cells. And other than that, they also make the retinal cells, the retinal neuron cells that I have mentioned to you. So all this that you can see here is to target on repairing spinal cord injury and also to improve visual functions, especially in the Stargardt macular dystrophy and also the myopic macular degeneration. Other than that, the embryonic stem cells has also been used to make cells to replace, to reduce the severity of the type 1 diabetes mellitus and the heart disease and also Parkinson's disease as well. So currently, the result shows that there are improvement in the patients in all these diseases, but the number of the patients that they have used is low. So there are more clinical trials that needs to be done in the future using a larger population of patients. So what is the ultimate significance in stem cell research? Okay, just now I've mentioned to you about making cells. How about if I tell you that if we can make these different kind of cells to form tissues and we can combine these tissues to make organ, wouldn't that be much more attractive to you? Isn't it? So worldwide, the number of patients that needs organ transplantations exceeds far a lot compared to donors from the disease and also from the living one. So there is a shortage. There's a shortage of the organs for transplantation. So currently, the type of organs that has been transplanted includes the heart, liver, kidneys, and lungs. So organ transplantation is desired, but at the moment still is not able to give, not able to produce the whole organ for the transplantation somehow. But one very good example of organ transplantation that has been successful using the stem cells is the bone marrow transplantation. So this work was pioneered by Professor Edward Donald Thomas from London, who is considered as the father of the bone marrow transplantation, and he won the Nobel Prize in 1990. So before 1960s, bone marrow transplantation was done, but not very successful. So he established the method to ensure that the cancerous cells in the bone marrow are killed by radiation first. So once the cells has been killed by radiation, then he transplant the bone marrow by intravenous method. So since 1969 to 2012, more than 1 million patients with otherwise fatal blood disorders, for example, leukemia, have received bone marrow transplants. So now, I have explained to you about the significance of stem cells that includes cellular therapy and organ transplantation. You might ask me the question, how can cells actually do the reparation in our body if it is given intravenously? So basically from the studies, from the literatures, we found that the stem cells not only able to differentiate, okay, by, in the lab, the cells can differentiate. That means you can induce the cells to differentiate in the lab. But if they are injected in our body, it is found that these stem cells can actually home to the tissue site of injury. So that means if your hands have an injury and if you induce in the IV, it will go to the site of injury. And then with the cytokines that is released from the dead cells after the injury, the cytokines will be able to induce the stem cells to form the cells to repair the injured tissue. So other than that, it is believed that stem cells also is able to communicate with the adjacent cells. So they are able to communicate by releasing proteins to the adjacent cells and ask them to probably to repair the cells by enhancing the survivability of the cells or to immunomodulate the tissue site. So this is a slide to show you about the mesenchymal stem cells. So they do have been shown to have paracrine effects. So they can release many proteins that is involved in the immunomodulatory, and also to repair the tissues, probably by synthesizing some cytokines that is involved in increasing the survivability or to reduce the apoptotic activities in the tissue site. So basically, we know that cells can uptake any molecules from the extracellular surrounding of the cells. And after the uptake of the molecules, the cells will respond and synthesize certain proteins and then excrete these proteins. So these proteins can be harvested in the forms of exosomes. So in the stem cell research, nowadays, exosomes is quite a hot topic now, because when we culture the stem cells, we can actually isolate the supernatant of the cell culture, and then we can centrifuge them or filtrate them so that we can concentrate the amount of exosomes and deliver it into the patients. So this method of treating the patients is advantageous because it will bypass any issues related to immune compatibility, tumor regenicity that can arise from the stem cells, and the transmission of infections associated with cell therapies. Other than that, it can also reduce the time and cost associated with the expansion and maintenance of clonal cell lines. So, other than that, we can also prepare the exosomes in very large quantities in an allogenic or off-the-shelf fashion and be immediately available for treatment when desired. So you must imagine if a patient has, for example, have COVID and the lung is damaged. So can we actually make the stem cells, induce them into lung cells and then transplant it? So this process will take a very long time. So if we can prepare the exosomes and immediately give to the patient, that it will be much more superior than using the cellular therapy. So there are also many ongoing clinical trials. In fact, about 20 years ago, many researchers have been focused on the study of using the cell directly to be infused into the patients. But now, if you look at all the researchers, so there have been a deviation. They are now looking at the exosomes to see how they can repair the tissues. So the ongoing clinical trials has been focused on to study the ability of the exosomes to treat dry eye syndrome in macular holes in China, COVID in China, Russia, and US. Exosomes has also been used to treat diabetes mellitus type 1, periodontitis, and Egypt, and also to use to treat multiple organ failure of aortic dissection in China. Other than that, because the exosomes from the mesenchymal stem cells, which has been believed to have immunomodulatory factor, so it has been used to treat dystrophic epidermolysis bullosa, which is a type of skin disease. So also, exosomes has been used to treat neurodegenerative diseases, including Alzheimer's disease and cancer as well. So this is what I meant with using the exosomes to treat COVID. So if you look at the clinical trial when the COVID happens, so at that time I was in Saudi Arabia and I was searching for the internet. So it was quite surprising that in the Middle East they are also quite advanced because they can find a way, they harvest the exosome of the stem cells and harvest exosomes from the mesenchymal stem cells and try to deliver into the patients to see how well they can reduce the inflammation in the lungs that has been infected by the viruses. So it has been found the mesenchymal stem cells can exert its reparative mechanisms through the exosomes, which has the anti-inflammation and immunomodulation activity. It can also repair the alveolar epithelium and endothelium because the exosomes contains a lot of factors. For example, the platelet-derived growth factor, insulin growth factors, hepatocyte growth factors. Many of these growth factors can help to induce the cells to make new lung cells. So I have finished my talk about making cells from the stem cells. I also have finished with the talk about using the stem cells to make the organs. So now I want to touch a little bit about making the stem cells to make organoids. Okay, so the stem cells can also be used to make the organoids. So here I show you a protocol which is taken from the neuroscience lectures. So in fact, there are many protocols depending on the labs. So here the starting material is to use the embryonic stem cells. They are being triggered to form the EB and then added with different kind of differentiation mediums to make the cerebral organoid. Sorry, how much time do I have? You can get 15. Okay. Okay, so now this picture shows you a cartoon image of the organoid. So when they are immature, this is how the organoid will look. But as they mature, they will form organoid in which the progenitor cells will arrange themselves in this area, and then they will start to become mature. So these cells will then migrate and delaminate from this layer, and they form the much more mature neuron cells. So in the literatures, it was found that the protocols, the many kind of protocols that has been established, managed to form the right kind of composition and also the percentage of neuron cells in the organoid compared to a real cerebral cortex of humans. So the organoids and also the combination of pluripotent stem cells is very helpful for us to develop a personalized treatment strategies to patients. So we know that there are about 7,000 rare diseases in the world, and we cannot give one type of drug to suit for every patient. So therefore, we are moving towards personalized medicine. And to illustrate to you, we can use the technology that has been well established. For example, we can take the patient's somatic cells and then make them to become pluripotent stem cells, and then induce them to become the brain organoids. So then the brain organoids that has been established can be used to compare with the unaffected or the healthy individual's brain organoids. Then from there, we can understand about the molecular events that happens in the organoid development, and also to look at what are the best drugs to give treatments to the patients. So therefore, this helps us in developing a personalized treatment strategies. So this is about the clinical application of stem cells. I'm almost done with my presentation. But I also want to bring to your attention about human evolution. A very nice story I got here. So this is a publication in 2021 in US in which they have used the organoids. They have used the organoids technology. So what they do in this study is that they wanted to see the differences between modern humans and also the Neanderthals. So we know that humans are much more closely related to Neanderthals other than other living primates like chimpanzees. So in fact, throughout the history when human evolved, we actually lose about 40% of the genes. Sorry, we lose about 60% of the genes. 40% genes is still contained in modern humans. So this group of researchers, very interestingly, so they take an archaic variant of a gene, which is called as a NOVA1 gene. So this is an ancestral gene, and they produce two brain organoids. In one brain organoids, they introduce this ancestral gene into the organoids, and they compare the development of these organoids. Interestingly, what they found out that when they inserted this ancestral gene, the brain organoid that they have developed can mature faster and have got a smaller size and different texture on the surface. Whereas in the organoids that is established from modern humans, the organoids grow slower and achieve the maturity slower. And they have got a larger size and have got a very smooth surface. So what they found out about from this study is that if you look at the chimpanzee, the baby chimpanzee can easily outsmart a newborn of a human. So, and they become mature faster than the human newborn. And then we have to teach the human newborns to be independent for some time. But we don't have to do that for the chimpanzees. The mother don't have to do that for the baby chimpanzees. So the results that they have got is corresponding to what they have observed in nature. So I have been talking about the promise or the potential of the stem cells, but I also don't want to leave the lecture without touching about the stem cell frauds and about the shonky business that's related to the stem cells treatment. So stem cells is a very hot topic in the past 20 years, and it attracts a lot of attentions, money, and prestige in the scientists. So in 2004, Professor Hwang Woo-sok, who is from Korea, has received the appointment as a supreme scientist because of his discovery to find a method to clone human by using somatic nuclear cell transfer, similar way Dolly has been cloned. Unfortunately, the 11 embryonic stem cells that's derived from the human is actually fabricated and falsified. So he has been dismissed from his institute. And then in 2014, Haruko Obokata in Riken in Japan has reported a remarkably easier way to induce mature human cells to form the embryonic stem cells by just simply dipping them into a weaker acid. And again, this makes a very a headline. So Dr. Haruko Obokata was also dismissed from Riken. So now, the latest publication in the British Medical Journal has debated about another surgeon, which is called as Paolo Macchiarini. So Paolo Macchiarini is a thoracic surgeon. So what is interesting about this story is that Paolo Macchiarini has tried to use diseased windpipes, which is a trachea, from the diseased donor. So he has used this windpipe, he has removed the cells and seed it with the patient's stem cells onto it. So other than using the diseased windpipes, he also used plastic windpipes. So he foresaw that organ transplantation using living or dead tissues is a thing of past, and he thought that he can use the plastic to make the organs. So what he did is that he produced the synthetic windpipes and seed it with the stem cells and transplant into 20 patients throughout this time in several countries. The bad thing is that none of the patients has survived. About 20 patients, and 20 patients died in a year after his transplantation. So his study actually is a scientific misconduct. It has caused a medical tragedy, and in fact has claimed many hits in the Karolinska Institute, which is a home to the Nobel Prize. So now Paolo Macchiarini has served has been sentenced to serve 2 years prison. I'm sure many of the audience here also realize that there are many springing of stem cell clinics worldwide, including in Malaysia. So the reason is because stem cells has given so much promise. So the clinicians and the publics know something about the stem cells, but they don't know everything about the stem cells, including the risk. So we have to understand that there are a few things that we don't know and which is not covered in this lecture, probably another opportunity. So that there is something to lose when you try an unproven treatment. Different types of stem cells serve different purposes in the body. So we cannot use the adipose stem cells for transplant into the retina. So we have to understand the development of the stem cells and which lineage of cells that they can develop and how they can be applied clinically. So the same stem cell treatment is unlikely to work for different diseases or conditions. The science behind a disease should match the science behind the treatment. Cells from your own body are not automatically safe when used in treatments. So patient testimonials and other marketing provided by clinics may be misleading. An experimental treatment offered for sale is not the same as clinical trial. So there are still so much more to learn about how stem cells work in the body and the capacity for healing. If the problem with the stem cells therapy offered directly to the public and it is not well regulated now is that it will hurt your bank account more than the disease will hurt your body. And before I end my talk, I also would like to take this opportunity to introduce you a book which has been co-authored and edited by Professor Suresh Kumar, Professor Akon Higuchi, and me, myself. We produced this book known as the Stem Cell Laboratory Techniques, a guide for researchers and students. So in this book, we have mentioned a lot. We have defined about the stem cells and many techniques that is relevant for you to culture the stem cells to and also some of the methods, for example, CRISPR-Cas, and also to confirm the quality of the stem cells here. So you can get this book from this website. So with that, I thank you for your attention.
Thank you, Prof. The floor is open for questions.
I think I'll take you back to 2004. You know, the medical science is such a way, I just put it as positive. 60s, we were working on prostaglandin, and we developed many drugs, NSAID and other drugs, prostaglandin analog. Because there are so many students out there, that is what I'm saying. 70s, we were working on a nitric oxide. People got a Nobel Prize also. Later, I met a person in IFOR meeting who discovered endothelin. So we could manage to get some of the drug, for pulmonary hypertension. Later, if you see that people are looking for a stem cell nanotechnology. Now, today's era, if you say that, another 20 years, you're going for artificial intelligence, machine learning. So this is kind of a moment it goes for 1 decade, and doing that, if you say, I remember 2004 and 5, we started working on stem cell. And typically, we decided to go for Burzer's disease. So the student, you can read the surgical book. If you get Burzer's disease, they say either you have a cigarette or you have a leg. So that kind of statement is written in Belly's surgical book. So we thought we'll give a GCSF, then we'll take the blood and we process it, we'll prepare a stem cell and we'll inject into, these calf muscles. And we could find dramatic improvement. People who cannot walk for a short distance, they started working. So since we have a control group and other, so in the OPD when they come, they said that these people are working, then they could find, though it is a double blind, it was something like that, there is a kind of improvement. And later they said that I want to have this kind of medicine also. But over a period of time, long-term follow-up, what you could find that it came back to baseline. So that is one thing is there. So then we did not do any clinical trial because many times you get very dramatic news that spinal injury, by giving stem cell, it has been improved, but it was actually compressed injury. So when you go for decompress, automatically it become improved. So as you mentioned, there are a lot of falsification of the data. So one should critically look at kind of therapy. So that's how, I was calculating the price. People come for a treatment, they put 22,000 rupees per injection, and it's kind of a placebo.
I think 22,000 is cheap.
22,000 ringers. So it's Indian currency is maybe 4 lakh rupees.
Means 400,000.
Yeah, so that is why it is very important that those who work right now, we are working on Alzheimer's and stroke. We are looking for stem cell derived growth factors. And we are doing only experimental study because we are not going for a clinics. Once you have a sufficient evidence, definitely will do. So I appreciate your, lot of concern about the stem cell and how we should go forward. Definitely, we need a targeted therapy for this. Thank you.
I think it's quite hard for the scientific community to actually regulate because it's so rampant. The opening of the stem cell clinic is so rampant. In fact, like US FDA president, have been asked about how is it that we can regulate, but they said that, and I quote them that it's difficult to boil the ocean because it's so rampant.
Yeah, similarly for other countries also, it's difficult to regulate.
Yeah.
But there are a lot of court cases also going on because of claim or misclaim.
But it's a very interesting research area.
Prof, from my side, what are the unintentional adverse effects or any sort of things are reported already here in this unintentional effects that would be there?
Yes, there is. There is. Like for example, the use of the fat stem cells. And it has been transplanted, if I'm not mistaken, is in the eyes for visual dysfunction. And because of this usage, it can cause death to the patients. So that's the reason why sometimes people might be, especially the scientists, maybe they also argue about this, about whether the stem cells work or not. But I think it works if it is applied correctly and we use the right protocol. And we cannot apply, like what I said, we cannot, we started this for this disease, we cannot apply it for another disease. It has to be, restarted again. It also depends on the stage of the stem cells as well.
Major concern is a cancer, unwanted growth, if it is not targeted.
Yeah, unwanted growth.
Prof, are there any regulations regarding this in Malaysia?
Malaysia. Okay, Malaysia is a bit. It's actually the same for all the countries. Malaysia. We started the study. Myself and my prof. We started since 2001. That time, we are very new in the mesenchymal stem cells. In fact, we are the one who established the culture method. And we try to disseminate the knowledge and teach people on how to culture. And after that, until now, there are quite many stem cell clinics. If you go around and survey, many clinics are around, but I would say that it's still not very well regulated. But there is some society that also helps together in collaboration with the MPRA to help to establish a protocol or a guideline to help the stem cells scientists and also those clinical practitioners who believe in the stem cells and try to get license for the products. Because at the moment, I think that if you search in the internet, there is no proper guideline. Because I think it is still, despite that it has already happened for more than 20 years, still the research is new because most of the time we use the stem cells application for compassionate patient, not on any individuals who still have the chance to live long. I mean, we use it only for those patients who have very, very worse prognosis. So the data that is acquired is not sufficient to really justify the promising use of the stem cells.
Very interesting topic, Prof. I just want to know about exosomes. You told that during COVID treatment of lung inflammation. How was the result, Prof?
How was that?
Means the treatment, means with the,
treatment?
yeah.
I am not very sure, but if you find from the internet, I think
Actually, we published a paper in biomedicine because during that time we had, because I'm a task force member, we had a lot of deliberation. Should you use, especially VIP patient? They said that can you use the stem cell? So I pulled all the data globally. I published. Just look at my name. You get it. So it is not effective.
it's not effective or effective.
It's not effective. So it's that is why I immediately published it because there's a lot of claim, and I wanted to publish so that people go in the right direction. We did that.
Very nice for you to share your experience. Good one.
Thank you for a nice talk. I have a couple of questions. One is regarding exosome. You said the exosome can be used for therapy, yeah? But I also know exosome also responsible for cancer. Many people are talking about the exosome are settling between normal cell and cancer cells. How do you look at this between these kind of exosomes?
Okay. To answer to your question, I think it's very important for us to characterize what's the content in the exosomes, right? At the moment, there is a guideline for the exosomes. And you see, the problem is that these exosomes are being isolated from the stem cells. And these stem cells are being isolated from different individuals. And these different individuals also have different ages, very strange. So therefore, the quality of the stem cells might also vary. So it is very difficult for us to get a consensus. Like what is the composition of the chemicals inside the exosomes? Do you understand me? Yeah, it's very difficult. Like, for example, the results will vary. We can't really have agreement or what is the composition of this composition in the exosomes. So maybe some of the data, okay, that use this exosome can cause cancer. The other one is not. It can help to repair, but probably because the composition is different. So that's why I think it's important that when people report it in a journal, they should also characterize the profile of the contents of the exosomes.
Thank you. Another one is regarding the stem cell bank. I think at present stem cell bank is very popular. So when a baby born, we can collect the cell and we can store it. That is a point. Then your last statement is that different types of cells cause different types of stem cells for different purposes.
Not different types of cells.
I can say like this also. Yeah, I can say like that because, sorry, do you want to finish your question?
Yeah. So then my question is, so if different types of cells responsible for different purposes, how the people store the stem cells from the baby? I guess basically they collect the cells from umbilical cord and store. So if different cells responsible for different purposes, how they manage the banking?
Okay, I'm so sorry if I have confused you about this statement about different cells is for different diseases or what?
Different purpose on the body, something you mentioned. Last slide. The second point, yeah.
Different type of cells serve different purposes in the body.
Yeah.
Okay, so I think what I'm trying to to explain here is that there are many different cells in different tissues in our body. And if we look at laboratory data, basically all these stem cells, they can be induced to form any type of cells. But the issue is about efficiency. So now I'm addressing about efficiency.
So then the stem cell bank, usually they collect the cells from umbilical cord.
Stem cell bank, yes, you can say so. Last time, umbilical cord.
Yeah.
Because in the umbilical cord, you can find the hematopoietic stem cells, and the tissues itself, you can find the mesenchymal stem cells.
I think it is going to be the huge business. So they are charging so much to storage, long-term storage. Okay, thank you.
It is like a bank you have to get the interest. We have the system. We created a core center.
I think we are not only having the blood bank or the stem cell bank, but we also have tissue banking as well, or organ banking.
No, it is biobanking now.
Yeah, biobanking.
Biobanking. So it will be good all.
Continue to that as a layman's question. So, as you said, Yamanaka introduced that induced pluripotent stem cells. If that is having more promising, so is it needed all these embryonic stem cells? Because induced pluripotent stem cells can exclude the possibility of rejection and can take immediately somatic cells and convert into pluripotent stem cells. So what is actually, how is going on? Because more than a decade actually induced pluripotent stem cells. Is any promising or what is the disadvantages of that?
Okay, I totally agree with you. That time was very hot, 2012. And then we started to learn the technique and we started to organize the workshops as well to teach people on how to make the induced pluripotent stem cells. Okay, the technical challenge is, because you asked me how is the results or how the outcomes, what is the status of the induced pluripotent stem cells. I think the challenge is to make this iPSC is already established. There is an established protocol, but the percent of efficiency is low. So maybe 0.few% of the cells that can be successfully induced to form the pluripotent. So, and then also the challenge is about technical challenge. Okay, to harvest the stem cells and to ensure that they maintain its pluripotency. So basically, I think Malaysia, the status is still not progressing much. But internationally, as you can see in the clinical trial, they do use the iPSC and make the retinal neurons for transplantation for improving the visual function. Yeah. So like me, the co-editor, Professor Akon Hikuchi, he's now in Taiwan. He is also involved of making the RP from the iPSC as well, and trying to transplant into the patients as well. But I would say that yes, there is some improvement in the visual function, and but the problem is that the sample of the patient is too small, so we need to do more testing on the patients, I guess.
Basically taking longer to
Yeah. But in the animals, you can see very good results, definitely. But we know that most time when we translate the preclinical studies to human, we are not able to really mimic or really replicate the results that we got from a preclinical study. I'm so sorry if I answered your question.
Yeah, correct. Actually, that because stem cells research itself very every invention is taking a long time. If you see the history also, after successful of the ES from animals, it took almost 2 decades, right, human to establish. So iPSC also maybe take some time. Once it's successful, I think many problems can be solved. At present, I have seen so many the tissues they are generating from the stem cell, but they are using for the research purpose.
Yes, only for research purpose at the moment. But I see that the trend is changing.
Hopefully, it's very interesting. Thank you so much.
That is because you can make an artificial vessel,
Yes.
artificial organ. So that's what this.
Yeah, at present that is successful, but in order to.
But how long it will last, that will depend.
That is.
Thank you for all the very nice
Questions and discussions.
One more question. Regarding the IPS, the induced pluripotent cell technology, is there any criteria for the cell harvested for this technology? Criteria for the cell which is chosen for this?
Yes. So first, after the injection, the cells, if they are being induced, they should behave morphologically like the embryonic stem cells, which means that the ratio of the nucleus to the cytoplasm will be high. If you look at the embryonic stem cells, so they should behave something like this, but you cannot see. Okay, so the stem cells, the embryonic stem cells, usually in a colony. Okay, in a colony, and they have got very smooth edges. They've got very smooth edges on the colony here. So we have to maintain, of course, we have to do the characterizations. We have to look at the markers, especially the OCT4, CMH, CLIF4, or the NANO. Those are the pluripotent marker. Other than that, we also have to, normally in research, we have to ensure that these embryonic stem cells are the true stem cells. So we have to induce them to differentiate into the 3 germ lineages, the ectodermal, endodermal, and the mesodermal lineages. They must express these markers before you can say that the induced pluripotent stem cells are pluripotent like the embryonic. So these are some of the most minimal characterization that you need to do. And the challenge is to ensure that they maintain its pluripotency, because if you do not attend to it, there is a tendency for the cells at the outer side to differentiate. So they can become any type of cells. And of course, we need to use a very specific type of media to maintain the pluripotency to culture them. And then we have to, then somehow we have to scrape the cells from here, and then we have to suck up the cells for expansion. I'm so sorry, did I answer your question? Thank you.
Thank you, Prof. Thank you for the very interesting and capturing keynote speech. Thank you very much. Second round of applause.
If you guys have question, you can ask her personally after this, okay? Okay. Thank you, Professor Dr. Madhu and Associate Professor Dr. Mok Pui Lim. I just want to remind all, if you haven't collected the goodies
Good morning, everyone. Today I'm going to do a presentation on illustrating the role of non-protein coding RNA, PMR 137, in the regulations of the virulence vector associated with the Fimbriae and also flagella in protease mirabilis through gene study. So this is my today's outline for the presentation. So protease mirabilis is a gram-negative bacteria best known for its multicellular surface mobility term swarming. It usually causes catheter-associated urinary tract infections, wound infections, urolithiasis, acute phenylnephritis, and so on. So the increasing multi-drug resistance of this organism poses a challenge to the scientific society to discover a solution for its pathogenesis. So in the year 2017, WHO released a list of bacteria which urgently require the antibiotic, including for the protein species. As you can see, protease mirabilis is a third-most bacteria which is highly isolated from the post-operative wound infection patients. So there are several virulence factors associated with the protease mirabilis like urease, hemolysis, motility and swarming, flagella and Fimbriae, proteinase, and so on. Fimbriae is a proteinaceous supermolecular structure in which it has the ability to adhere to the host epithelial cell. Flagellum is a motility organelle that enables the movement and also chemotaxis in protease mirabilis. It also helps in the protein export and also the adhesion, which are contributing to the pathogenesis after the invasion of the host cell. So what is NPC-RNA? NPC-RNA is a non-protein coding RNA, which also known as a small RNA transcript that present between 50 to 500 base pairs. The bacterial NPC-RNAs were recognized and associated with the regulation of replication of extrachromosomal elements. The regulatory NPC-RNA is shown to play a key regulatory role in diverse biological processes. For instance, like the modulation of transcription and translation, mRNA stability and DNA maintenance to silencing, comprising changes in RNA conformation, protein binding, and so on. So SLHD is a transcriptional activator that binds to the promoter region of flagella oblong and activates the transcription of the oblong, which leads to the synthesis of protein. These transcriptional activators show both positive and also negative regulation when it is directly pairing up with the NPC-RNA in E. coli. So these indicate that the NPC-RNA is actually playing a crucial role in the virulence vector of the bacteria. NPC-RNA PMR 137, which is one of the novel NPC-RNA from protease mirabilis, were identified through the unpublished data funded by the FRHS. So these NPC-RNA were selected in this gene knockout study. It is because it is highly expected to regulate the flagellar transcriptional activated protein encoded by the FLHD gene, which is associated with the bacterial virulence vector. So the objective of this study is first to knock out the NPC-RNA PMR 137 from the protease mirabilis genome, and then to sequence and analyze the transcriptome of both wild type and also the mutant strain, and then to understand the differential gene expression of NPC-RNA of both wild type and also the mutant of protease mirabilis, and confirm their expression under various stress conditions using the northern blot analysis. So the methodology we started with the single-step gene knockout of PMR 137 and insert the kanamycin cassette into the PMR 137 location of protease mirabilis. And then we extract the total RNA from both wild type and also the mutant strain and send for transcriptome sequencing and run the transcriptome analysis, and confirmation of the last part is the confirmation of differential expression analysis of novel NPC-RNA. So we have done the transformation of kanamycin acid together with the homologous specific region into protease PMR 137 location in protease mirabilis cell, and we screen it on the kanamycin plate. As you can see, there are several colonies were grown on the kanamycin plate. So in order to verify the absence or the knockout of PMR 137, we proceed for the PMR 137 gene amplification. As you can see, one of the colony did not show band near the expected basis. So these indicate that the colony is absent to the PMR 137 gene. These genes will proceed for the verification of the insertion of kanamycin resistant gene, which is the kanamycin cassette together with the homologous specific region in the PMR 137 location. As you can see, the wild type strain shows band near 300 base pair, while the mutant strain shows band near 1500 base pair. So these indicate that the kanamycin together with the homologous region was successfully inserted into the PMR 137 location. So further verification was done for the with the transcriptome sequencing, where here we have found total around 224 peaks were identified. The peaks were screened using the BLAST, R-time, and also the ORF. So from that, 132 candidates were selected as a possible novel anti-Zionist, while 92 candidates were recognized as annotated genes. So from that 132 candidates, 65 genes were upregulated, 66 genes were downregulated, and one gene was not differentially expressed. So this is an image shows the MA plot showing the differential expression of the anti-Zionist. The dots that falls above the red line indicates the upregulated genes, anti-RNAs, while the dots that falls below the red line indicates the downregulated anti-RNA genes. So from that, I have selected 5 anti-RNA based on the target mRNA prediction.
there is a greater need for the production and the novel drug that is needed to combat these. So the lack of specific antiviral treatment was known, and viral replication and intricacies. Antiviral drugs, the virus has a property to mutate and replicate itself from the host. Mutation makes it difficult for us to provide antiviral vaccine, and the time constraint during this pandemic was a study or in the based on the target study. So these are the targeting the main protease of the SARS-CoV-19 is viral replication and the protease function. The virus to replicate and produce a new viral particle, it must undergo a series of complex in the host cell. The main process, also known as this 3CL Pro, is responsible for the cleaving of the large polyprotein. So disruption of the viral replication indicates this. The cleavage and the disruption of this viral polyprotein makes sure that the enzyme and main target protein of us in the SARS-CoV-2 is being stopped, so that the mechanism of replication on the mutation does not happen in this section. The broad spectrum antibiotic is of major impact for this specific target protein, and the target design, three-dimensional structure of the protein was protease SARS-CoV-2 was they derived from the UniProt database, and the retrieval of the ligand from the impact database was done, and the preparation was done using the molecular Bionova Discovery Studio. The virtual screening of this ligand and the structural protein was done in the Pyrex software, and the suitable ligands were selected based on their binding entry and the scoring function. And the methodology is the accession or the target protein was retrieved from the PDB database. The X-ray crystallographic structure was available in the structure, so homology modeling was not required in order to template the structure. And then the interaction is particularly with our target protein and the ligand. The molecular structure of the protein was prepared, and the virtual screening, the Pyrex software, 300 impact the ligands were retrieved, and then they were docked, and the scoring function was the major point to select the ligands that had a higher binding energy. And the virtual screening, based on the virtual screening, the selected ligands were selected based on the binding energy, the ADME properties, and the target protein of attack using these softwares. And there was in these 300 compounds docked, 7 compounds were of our lead candidates because of they had a best binding energy when compared to the control agents that was used. And this is the control agent. The two control agents we have taken in order to specify our study with this result is the hydroxychloroquine, which had a binding energy of minus 5.7 kilocals, and another control that we had used is acetaminophen, that is minus 4.9 kilocals, with the target protein, that is the main protein SARS-CoV-2. With these two targets, when we had the adopt our ligands of the selected lead compounds, they had a better binding energy of 9.2, and this is the lead compound that we have selected, which had a binding energy of minus 9.2 kilocals, and the second lead compound had a minus 9 kilocals when compared to the control agents. Where these two had a higher binding energy, and it has a better attacking
better ability to attack the target protein. So my conclusion here in the study presents that the lead compounds have a better binding energy and a better candidate for a novel drug when compared to the available drug compounds that we have been using, and which had this binding energy of minus 9.2 and 9.9. So the best, according to theoretical result and the computational work, this present study shows these lead compounds have a better identical study when compared to the available control agents. Uh,
Uh, any questions from anyone?
So what is, excuse me, thanks for presentation. So with your findings, how are you going to verify your results in the laboratory?
Okay, Miss Rachana Sri.
yes, sir.
So after this in silico identification, how you can validate in either in vitro or in vivo conditions?
In vitro study, there is antibacterial screening work that can be done, and molecular studies work that can be done in order to facilitate the mechanism of study, in order how it works, how it is being isolated, and their extract can be used in the in vitro studies.
Okay, thank you, Ms. Rachana Sri.
Thank
So
you.
then we will go to the last presenter. Before last presenter, I would like to see once again any Miss One attending now. So I'll go through from the list. Atista is around.
Yes, sir, I'm here.
Oh, Miss Atista is here. Okay.
Yeah, yes, sir.
What about Sayeda? Kritika? Jayashri? Manoj Kumar?
Yes, sir, I'm here, sir.
Oh, Manoj is also here. Okay, then Sushmita?
Yes, sir, present.
Selva Vensi? Okay, so before we go to these three who didn't come from the beginning, so we will go to the first, the last speaker supposed to be, then we will go to the previous one. Okay, so OL17 Hema. Hema is on.
Yes, sir.
Judges, the number is OL27, please. 27. Yeah. Okay, go ahead, sir. Hema.
So we are from Vivekananda College. We have group of 4 members. Can you present one by one, sir? And the same topic.
But we can give only 7 minutes.
Yes, sir.
Okay, go ahead. Share your screen, please.
Yes, sir.
Good morning, everyone. Me and my friends are present on the topic of fabrication of humic acid beads from water hyacinth for the absorption of water contaminants from the College of Arts and Sciences, Namakkal. Moving on the topic,
Can you minimize the small icon window from the from the Teams? Can you able to?
Yes, sir.
It's blocking your screen. Ms. Hema, can you once again share your screen?
Okay, sir. Is it visible, sir?
Yeah, visible, visible. Okay. Uh, change
Should I
the screen to
start?
the landscape. You rotate the screen. Okay. Go ahead.
Okay, once again, good morning to everyone. Me and my friends are present on the topic of fabrication of humic acid beads from water hyacinth for the absorption of water contaminated from Vivekananda College of Arts and Science. Next, moving on the topic, rivers, reservoirs, lakes, seas are nowadays drowning in the chemical waste, plastics, and other pollutants. This problem of water is spread to our health because water is an universal solvent. There are many methods and ways are followed to remove the pollutants and other contaminants like heavy metals. Phytoremediation is one among the methods to remove the contaminants. Phytoremediation is a method which is using the living plants to clean up the soil water with a hazardous compound. For this purpose, we are using humic acid. Humic acid is an organic matter formed from Hema. Humic acid is an organic matter formed from a decaying plant and animals before it becomes to a lignite. One of the main properties of the humic acid is a chelating property. In this study, humic acid from water hyacinth is combined with the sodium alginate beads treated with the polluted water and analyzed in the UV visible spectrophotometer for the characterization of heavy metals and absorption. Next, aim and objective. To isolate the humic acid from water hyacinth plant and to analyze the heavy metal absorption. For the sample collection, water hyacinth is collected from a Kumaragiri Lake, Salem. The leaves and roots are dried under the shade under the room temperature of 48 days and the samples are grinded from a fine powder. The next slide will be explained by Hema. Thank you.
Good morning to everybody. The plant was powdered and dried for over days. Then, is my voice audible? The powdered sample was allowed to operation in the proportion of 1:10 for the oxalate method. The humic acid is obtained from the acidification. So we have used heptone and a methanol in the ratio of 2:1. Then it was allowed for overnight, and this is the solvent obtained from the oxalation. We have treated it with 0.1 ml of NaOH, and then it was centrifuged, and the supernatant was allowed to react for HCl for the acidic condition. Then the humic acid was allowed to precipitate for 2 to 3 days under the shade. Then we have move on to the preparation of sodium alginate beads. So for the enzyme entrapment method, we have prepared also preparation of humic acid beads in comparison to both in comparison with the pond water. Then we have taken 3 conical flasks named as first one of S1 containing of a pond water and S2 of pond water along with the sodium alginate beads and third tube of humic acid beads. The sample was left for 24 hours. 3 ml of sample was the 3 ml of sample was taken from the tubes and observed under the UV visible spectrometer. Then the same beads were left over 24 hours with the refillment of 3 ml of pond water, and we have observed it after 1 day. Then the result was in the screen. In the S1 tubes, we have observed 3.15, and pond water with the sodium alginate beads of 0.22 absorption, and pond water of with the humic acid beads of 0.18, and a second day for same beads along with the refillment of pond water. The pond water is of 0.3, and the pond water with sodium alginate beads of 0.180, and pond water with the humic acid beads is of 0.046. This shows the graphs of, we have observed the UV visible spectrometry from the 220 to 700 nanometer, and the range is greater than 360 nanometer. In conclusion, we have isolated fumic acid from the walls and hyacinth and isolated fumic acid walls, and to observe the increased surface area of the absorption, treated on the water sample to analyze the absorbent capacity of the fumic acid in removing the heavy metals like cadmium ion. Then the analysis in the UV visible spectrometry is a high peak at 267 nanometer. This represents the efficacy of the fumic acid in removing the heavy metal. Thank you.
Actually, the methodology is not clear for me.
So the comment from the judge is the methodology is very not clear.
I think we can say that it is effective or not effective. It's supposed to have experiment again and control and see what's the difference. By this way, we can see effective or not.
Yeah. So the based on the results and methodology at present, we cannot clearly can say whether it is affected or fully not affected. Maybe we need to do the further study. That's a comment from the judge. Okay, thank you, Ms. Hema. Then we will go to the one which is missing from the list. Atista, quickly, can you present?
Yes, sir. Shall I present the screen, sir?
Yeah, this is OL1, Mr. Atista.
Yeah, yes, sir. Okay.
OL1.
Okay, a pleasant good morning to all. I am Adista, here to present my studies on computational analysis of bioactive compounds from Diospyros ebinum, Oldenlandia umbellata, and Momotica cementia as promising inhibitors of HMG-CoA reductase involved in hypercholesteremia. So, begin with hypercholesteremia, generally characterized by elevated level of cholesterol in the bloodstream, and its subsequent accumulation may causes cardiovascular disorders like heart attack, stroke, chest pain, and atherosclerosis. And this is the general pathway involved in cholesterol synthesis. And in this pathway, there is an enzyme called HMG-CoA reductase, which converts HMG-CoA to mevalonate. Mevalonate are the precursors for cholesterol synthesis, and this is the rate-limiting step which determines the overall rate of cholesterol synthesis in the body. So, to combat the level of cholesterol in the body, the inhibition of HMG-CoA reductase or reduce the activity of HMG-CoA reductase is highly beneficial. So there is a drug in the market, commercially available drug called statins. The statins are commonly called as HMG-CoA reductase inhibitors. There are different types of statins in the market like simvastatin, mavastatin, etc. This is a widely prescribed class of medication for managing high level of cholesterol in the body. However, this drug have a highly efficient activity to reduce the level of HMG-CoA reductase in our body. So, however, as a chemical drug, this statin poses some side effects like insomnia, abdominal pain, and also a liver failure over a time. So, we can use natural compounds as an alternative to this type of statin drug for managing the high level of cholesterol in our body. So this study mainly focused on natural plant-based compounds as potential inhibitors for HMG-CoA reductase. So for our study, we have chosen 3 plants. that is Diospyros sabinum, Oldenlandia umbellata, Momotica charantia. These three, from these three plants, Momotica charantia, commonly called bitter gourd, bitter melon. This bitter gourd holds a promising activity towards hypercholesteremia and hyperlipidemia and obesity. But apart from this, Diospyros sabinum and Oldenlandia umbellata has not been much more studied towards obesity or hypercholesteremia. But this Diospyros sabinum is commonly called Ceylon ebony or Indian ebony, and this Oldenlandia commonly called as chai root. These plants possess an antioxidant, anti-diabetic, and hepatoprotective activity. So we have chosen these plants also for our study. So this as a potential inhibitors for HMG-CoA reductase. So the main objective of our study is to retrieve the bioactive compounds from Diospyros sabinum, Oldenlandia umbellata, and Momotica cinerea, which is completely through a database called IMPACT and also through an extensive literature survey. And to analyze the pharmacokinetic and toxicity profiling of the bioactive compounds to determine the efficiency of these compounds. To determine the interaction of bioactive compounds with HMG-CoA reductase, which is a key enzyme that serves as a therapeutic target for treating hypercholesteremia in our body. So the final step is to identification of potential HMG-CoA reductase inhibitors among all the bioactive compounds which is obtained from these three plants. These are the methodology. For target identification, we have used UniProt. We retrieve the structural and functional information about the target from UniProt PGT, and we retrieve the for ligand selection. We used IMPACT and through literature. And then we identify the target binding site. Target binding site, which is highly necessary to know where our compound should bind and inhibit the activity of HMG-CoA reductase. Then we analyzed the pharmacokinetic property by using Sysdme, and we did toxicity profiling of those compounds using Waseris. After all these steps, only the selected compounds which satisfy the Sysdme and Waseris criteria, we did docking with HMG-CoA reductase. This is the target information that we have chosen. This is the complex structure of a HMG-CoA reductase with mevastatin. It's a drug. So analysis of binding sites. Initially, we started analyzing binding sites by docking HMG-CoA reductase with a drug, the statin, to know where our compounds also should bind and inhibit or reduce the activity of these compounds. From that, we analyzed that 690, lysine 692, lysine and serine 684. These are the residual and important amino acids that plays an important role in inhibition of HMG-CoA reductase. And next one is slide, and we also used a database called TrankWeb. We used that for ligand binding site prediction, and these amino acids are relevant to each other. This binding site also shows relevant amino acids where our compounds also need to bind in that region. So ligand identification is completely through the IMPACT database and extensive literature survey. For in this study, we identified 14 compounds from Diospyros sabinum and 15 compounds from Oldenlandia, 222 compounds from Momotica charantia. Altogether, we selected 252 compounds for our study. So these are the list of compounds present in Diospyros sabinum and Oldenlandia umbellata. And these are the 222 compounds present in bitter melon. System pharmacology, we used Sysdme. For every drug should have some criteria to become a lead molecule. So we analyze the physicochemical properties and its efficiency of a drug molecule by using Sysdme by considering the following parameters like molecular weight, drug likeness, hydrogen bond acceptor, donors, and if the compound have any lipid violation, and also measuring the bioavailability score. After these pharmacological studies, the compound get reduced to 79 compounds. Only 79 compounds have the ability to become a lead molecule. Then we did toxicity by using Waseris. This Waseris tool helps us to know whether the compound have any mutagenicity, reproductive effect, and tumorigenicity. So from these Waseris studies, it helps us to know that how many compound doesn't have any violation against these properties. So from that 79 compounds, only the 29 compounds has fulfilled these criteria, and this 29 compounds is completely from 27 compounds, which is from Momotica cerantia bitter melon, and 3 compounds, two compounds, that is ebinone and ursolic acid. This ursolic acid is present in both Diospyros ebinum and Oldenlandia umblata. So these are the results: molecular weight and rotatable bond of all compounds. Finally, we have chosen ebinone and ursolic acid from Diospyros ebinum, and ursolic acid from Oldenlandia umblata, which satisfy all the criteria of pharmacology and toxicity analysis. And these are the 27 compounds which has potential activity towards to become a lead molecule. So after that, we did docking by using a tool called LibDoc in Discovery Studio. These 29 compounds we have docked with HMG-CoA reductase. In this 29 compounds, all the compounds almost binding with the target efficiently in the same place where statins used to bind. And all the 29 molecules except biocyanin A and riboflavin, these 2 compounds present in bitter melon, and it has one unfavorable bond, that red bond with HMG-CoA reductase. And this is the novel compound, that is ebinone and ursolic acid. This ebinone is present in Diospyros ebinum, and ursolic acid, which is present in both Diospyros ebinum and Oldenlandia umblata. These compounds make the interaction with HMG-CoA reductase where statins bind, like 691, 684, and 561, etc. And these are the novel compounds that we have identified from those plants. And there is more studies like in vitro level towards bitter melon. So in this study, we analyze the compound level, how the compound interacting with this target. So in this, we identified these are the compounds from Momotica. It efficiently bind with HMG-CoA reductase. Like the serine aldehyde, luteolin, momodicin, it have a good binding interaction with the target, and it also have a good LibDoc score with the HMG-CoA reductive. In these, from the triple 2 compounds, these 2 compounds has not much more, doesn't have any much more studies towards hypercholesteromic activity. So hereby I conclude that I wish to conclude that this study may pave the way for developing natural remedies to treat hypercholesterolemia, prevent obesity, and other associated disorders. That will be it.
Okay, thank you.
Thank you for your presentation. I would like to ask, with the findings that you have, do you have any plan to do studies on the animals?
Mr. Sister, I repeat the question from the judge. So with the findings of your work, so are you planning to continue with any animal studies further?
Yeah, so further we have to do HMG-CoA reductase enzyme assay. The enzymes are already available, so we thought of doing HMG-CoA reductase assay, and also we further proceed like zebrafish animal studies, gene expression level.
Is it possible to create a hypercholesterolemia model in the zebrafish?
Is it possible to make a zebrafish with a hypercholesteremia model?
Yeah, sir. Actually, almost more studies are there.
Okay, good. Thank you.
Yeah, thank you.
Okay, from your study, you said you tracked the bioactive substances from three acting microorganisms or something. So if there is a difference in the bioactivity of these 3, which is the thing you used in your study?
3 plants.
3 plants, yeah. Is there a difference in the bioactive compounds between the 3?
Hello. Is there any difference between the 3 different biomolecules which used?
Difference means, I'm sorry?
The 3 plants you used to drive the bioactive compounds from 3 plants, right?
Yes. 3 plant species, the activity differences.
Okay. You have taken the three different bioactive molecules, right?
Yeah.
Have you found any difference in the activity in your study?
In the interactions, yeah, sir. Almost this statin drug, it binds with HMG CoA reductase with almost 8 hydrogen bonds. So our study, if the APU binds with one amino acid, means other compounds have an interaction with another amino acid. So the hydrogen bond interaction may differ. Yeah.
So I think that's it from the judges and any questions. So thank you, Ms. Atista. Very nice presentation. Then we will go to the next one, Sayeda. Ms. Sayeda, are you there? Not there. Kritika? Kritika? Also not there. So then Jayashree? Jayashree? Okay, Manoj? Manoj?
Yes, sir. Yeah, yes, sir.
Okay, I think
Shall I present it?
this is the last one. We will not call once again others because some is missing. So this is supposed to be the last presentation. Anyone else not yet presented? Can you unmute and say anything?
Sushmita, sir.
Sushmita. Okay. You are after Manoj. Okay. So then it is last but one. So then after Mr. Manoj, you will be ready, Mr. Sushmita. So Mr. Manoj, can you share your screen, please? Thank you.
Sure, sir.
This judges, this is OL9. Okay, go ahead, Mr. Manoj. Share your screen, please.
So good morning, everyone. I'll be sharing my presentation on characterization of silver nanoparticles using the Cinnamon xylonicum bark as a potent antibacterial agent. So the silver nanoparticles, these are the tiny yet mighty nanoparticles that are getting everyone's attention worldwide. It is extremely important to find eco-friendly ways to produce them, especially for the medical purposes. These silver nanoparticles shows incredible power in fighting off different harmful microorganisms, which makes them very promising in the battle against disease. Our study focusing on plants to create AGNPs and that is the silver nanoparticles and testing their outstanding ability to kill the bacteria. So the next slide will be telling about the objectives of the study. So we are going to dive into the process of using Cinnamon xylonicum bark as an extract to make silver nanoparticles and explore its unique properties as a powerful bacteria fighting agent. I've also used different techniques like UV spectroscopy, FTR and TEM and XRD for the characterization to analyze the silver nanoparticles and gain valuable insights into their structure and properties. In meantime, we'll be also checking out the effectiveness, how they are effective against the various bacteria. So the next slide will be the exploring the potential of the Cinnamon xylonicum bark. So the bark method produces silver nanoparticles with a significant potential. The bark imparts essentially physicochemical characters to silver nanoparticles and holds immense potential in biomedicine. Its unique properties help create a potent agents and battling the harmful pathogens effectively. So the next slide will be the methodologies.
We cannot see the movement of the next slide, Mr. Manoj.
Just a moment, sir. I'll be stop sharing and again I'll be sharing, sir.
Okay.
I hope it is visible, sir. Huh?
Yeah. Go to the presentation mode and continue.
It is in the presentation.
We're not seeing in a presentation mode. Anyhow, go ahead. I think we are running out of time. Just.
Oh, sorry. Yeah. Yes. Yes.
Okay.
So this is the methodology we've been following. The bark of the cinnamon xylem was collected and underwent a meticulous cleaning process using tap water and distilled water. After cleaning the barks, they were diligently dried in hot water oven, and the bark was cut into small pieces and finely powdered to make evenly mixed. This was really important to the synthesis to be successful. After that, the 5 grams of cinnamon xylonicum powder was mixed with 125 ml of double distilled water. The mixture was heated to 70 degrees Celsius and boiled for 30 minutes. And the next slide would be the result and discussion slide. What I've done is I explored various concentration of bark extract to identify most effective conditions for producing silver nanoparticles. Actually, I measured 8 ml of cinnamon xylonicum bark extract with 2 ml of silver nitrate solution. There is a color change from yellow to brown, which means that successful made by the silver nanoparticles. And I put so I'll be sharing the next slide would be the images of that displays the transition of color yellow to brown following the addition of silver nitrate to the sample. So these are the findings of UV spectroscopy. The results represent the absorbent spectrum of our sample. Within the spectrum, there is a distinct peak stands out of 426 nanometer. The peak indicates the presence of silver nanoparticles. So these are the 10 images. And the results and the investigation and was spherical silver nanoparticles with striking with white and black dot pattern, measuring 2000 nanometer wide. Each dot represent the silver in our nanocomposites. The TEM analysis reveals that nanoparticles ranging from 5 to 20 nanometers in size. And the next will be the FTR analysis. The FTR in this there is a prominent peak at 2 centimeter, indicating the CS3 spacing and the methylene groups. And so this is a nanoparticle stability that is the zeta potential value. And the zeta potential value of silver nanoparticles extracted is minus 250 minus 25. So this is a nanoparticle structure that is a clady pattern reveals that the peak is close to 20, and this displays the silver nanoparticles are varying in crystallinity, providing the valuable information about the structural arrangement. And this slide tells about the antibacterial activity. The result shows that our synthesized AGNPs exhibit impressive antibacterial activity. That is, the zone is measured by that is 15 millimolar and 20 millimolar for 20 and 30 mule 30 mule. The pathogens which have been used is Escherichia coli and Staphylococcus aureus. And this is the image that shows results of the antibacterial activity using the disk diffusion method. In conclusion, our research focused on the cinnamon xylonicum bark extract to create silver nanoparticles with strong antibacterial properties. We successfully synthesized silver nanoparticles with an average size of 5 to 20 nanometers, showing the stability and potential use. These AGNPs demonstrate remarkable effective against bacteria with a zone of inhibition measuring of 20 millimeters. So that's all to the team, and thank you.
Okay, thank you. Thank you, Mr. Manoj. Any questions from the judges?
Yeah, we talked about the it's for very high effective or something. Can you go to the conclusion? Yeah.
Can you go to the conclusion, Mr. Manoj?
Sure, sir.
Is it exceptionally high? How we can define exceptionally high?
Yeah. See, if you see the last conclusion, you mentioned in the last line, you mentioned is found to be exceptionally high and showing a remarkable measurement. So, how you can say exceptionally? How you can define that?
Uh, the zone that comes around like for the is 20 millimolar. So that is like while comparing to the commercially available, that is exceptionally high. That is how I define it.
Okay.
I think you have some experiment to compare this and the commercially available.
Have you compared with the commercial one in your study?
Um, no, sir. Yeah. Just article review.
Yeah, that would have been better if you do parallel. We can see the zone of inhibition comparison. Of course, you have compared with that commercial one, but not the published one, right?
Yes, sir.
Um, Thanks for presentation. Um, is there any literature or any reports that has also used um, cinnamon silicon bark extract to synthesize gold nanoparticles? And what's the efficiency differences
Yeah.
when compared with silver nanoparticles?
Yeah. So, Manoj, is there any previous studies on this cinnamon gelanicum about either gold nanoparticles or silver nanoparticles, and how you compare with the previous study?
Like that, there was many paper regarding this gold and silver nanoparticles. The first, like we are planning to go on further studies with animal studies also, like efficiency of how our antibacterial study is going to be done. So that is one thing that will be done. And yeah.
Okay.
Yes.
Thank you, Mr.
Yes.
Manoj. Then we will go to the next speaker, Mr. Smita. Can you unshare your screen, Manoj? Susmita will share. Susmita, can you share your screen, please?
Yes, sir.
Ms. Susmita is OL10. OL10. Susmita, are you sharing the
Sir,
slides?
is it visible?
No, you are sharing the Gmail.
Oh, yeah, okay, sir. Is it visible, right?
Yeah. It is visible, but it is coming your Gmail. You can Gmail window we can see, not your PowerPoint window.
Oh. So how about now?
Still same. Maybe what you do, you remove you unshare first and share again the correct window. And I can see some Divya Christie raising the hand. Anything you want to say, Miss Divya?
So can you hear me?
Sorry?
Hello, sir.
Yeah?
Can you hear me?
Yeah.
So I have presentation for OL 8.
Sir, is it visible now?
OL8 is Jayashree, right? Jayashree, isn't it?
No, Divya Krishna. Yeah, it's my fourth corresponding author's name. Yes.
Oh, is it? Okay.
Yes.
So that's what we were given. The name is given was Colin. Okay, just wait for a while. Let's Miss Sushmita finish.
Okay, sir.
Mrs. Smita, go ahead. Put in the power presentation mode. Sushmita. Sushmita, can you hear us? We can't hear you.
Sir, is it visible now? Can you hear me?
Yeah, can hear. Okay. Can you quickly present?
Sir?
We can see the screen. Can you present? Start presentation, please.
Hello, sir.
Yes, yes. Ms. Sushmita. Now you already unshared. Can you share once again? It looks like you are having some issue with the sharing. Maybe can you solve the issue? We'll go with the Miss Divya first.
Yeah, okay, sir.
Miss Divya, are you ready?
Yes, sir.
Yeah, could you please share your screen?
Yes, sir. Good
Yeah.
morning, sir. Can you hear me?
Yeah, can hear, can see the screen, and you just go ahead.
Okay. The topic of my presentation is role of neuropeptides in dopamine neurogenesis. So the background of the study is, so normally the dopaminergic neurons regulate a diverse set of behavior from the control of movement to modulation of cognition. So it was found in the harsh region of the brain, that is the substantia nigra part of compacta. So if these neurons were degenerated, this caused dysregulation of motor control, hallmark of Parkinson's diseases. So the Parkinson's disease is the second most commonly occurring neurodegenerative disorder, where this mesencephalic dopaminergic neurons are responsible for the motor function and postural stability are degenerated. So this system plays a central role in several significant medical conditions, including Parkinson's disease, attention deficit hyperactive disorder, Tourette syndrome, schizophrenia, bipolar disorder, and addiction. So neuropeptides are small protein-like molecules used by neutrons to communicate each other. So neuropeptide Y, a strong oxygenic hypothalamic neuropeptide, which can alter the dopamine neuron activity to affect feeding. It is the most abundant peptide present in the mammalian nervous system. So this NPO gene might influence the development and function of NDA neurons. So in the current study, we aim to understand this NPY during the process of neurogenesis and degeneration. In order to characterize the NPY gene, we used MPP plus toxin to ablate the dopamine neurons. So zebrafish, why we have used zebrafish as a model system? Because it has a number of features that make an attractive research tool. And the fundamental advantage is that they share considerable amount of genetic identity with humans. And moreover, the translucent body of the embryos facilitate non-intrusive visualization of organs and biological process in vivo. So, in combination with this, zebrafish are genetically tractable, amenable to large-scale forward genetic appraisals. So, additionally, they are relatively inexpensive to maintain, produce large numbers of offspring, and undergo rapid development. So the aim of this project study is to determine the role of neuropeptides in dopamine neurogenesis using zebrafish as a model system. So in order to accomplish this, we have followed the following objectives, like to study the functional role of NPY during the process of neurogenesis and to analyze the nucleotide variation of this gene following a specific dopaminergic neuron ablation using neurotoxin NPT+, and then study the gene expression patterns during the process of neurogenesis and neurodegeneration using in situ hybridization, functional characterization of NPY gene during the process of neurodegeneration using qPCR. So this is the result and discussion. So phylogenetic tree examines the evolutionary relationship between
in the organism, the greater the length of the branch, the longer the period of time. So the tree shown is based on the alignment of 7 homologs. So in the Homo sapiens, Oryctolagus and Maca are closely related and have a common ancestor, while Homo sapiens and Macaca mulatta have a percentage similarity of 82% NPY gene similarities, while this Oryctolagus have a gene similarity of 82.4%, forming a clade, and hence they are closely related. While Gallus do not have a common ancestor, and it has a percentage of 86.5% of gene similarity. And Rattus norvegis and Mucus musculus are more closely related, and they have a common ancestor percentage of Rattus norvegis and musculus are 50%, which indicates that they share the gene similarity with the common ancestor. But in Danurerio, that is zebrafish, does not share close similarity with NPY homologs from the rest of the organism, which clearly states that they do not have a common ancestor and gene similarity with the common ancestor at 60% nucleotide substitution. So these are the mRNA expression of NPY gene at various developmental stages. So after continuous exposure of NPP plus at various developmental stages, that is 24 to 96 HPF, the expression of NPY gene was normalized with beta actin, and we have noticed that the expression was increased in control group with the developmental stages. This supports throughout the developmental stages that NPY gene expression is required to maintain homeostasis. When exposed to the neurotoxin NPP+, NPY gene expression was decreased significantly with a p-value of less than 0.05 at 48, 72, and 96 HPF respectively. However, at 24 HPF expression, the gene was not measured. It is an initial exposure, so maybe the activity of NPY gene may be completely demolished. So although the results were similar to the study where the dopamine neurons appear to stimulate NPY and SOM gene expression, dopamine excess dissimilar that leads to loss of production of dopamine. So altogether, the dopamine neurons lost their activity to the regulation of NPY. So this is the quantitative expression of gene. And next, we have done a sequence analysis, DNA sequence chromatogram, demonstrating the mutation at NPY gene. So in order to characterize the NPY gene, we have done sequence analysis. In this data, we compared the nucleotide variation at control, 96 HPF in control and one NPP plus group, and we observed few nucleotide variations. So these nucleotide changes may be due to the decreased expression of NPY gene. Next, in situ hybridization was performed only at 96 HPF, and we observed expression of NPY gene in the region of mesencephalon and telencephalon of the zebrafish brain region in control group. After ablation using NPP+, we observed the expression of NPY gene drastically decreased in the region of telencephalon and mesencephalonic regions. The decreased gene expression was correlated with the qPCR expression analysis. So from this, we can come to the conclusion that while inducing NPP+ at various developmental stages, there was significant reduction of NPY expression in mRNA. So to conclude, previously in our lab, we have administered MPP plus during the critical window of dopamine neurogenesis, and it was found that it will degenerate the dopamine neurons at the 96 HPF by 70%. So we were curious to know in what way the MPP plus affects the NPY gene expression. So interestingly, the gene expression reduced in time-dependent manner, which correlated with the loss of dopaminergic neurons. Similarly, the expression was also reduced in the mesencephalon and telencephalonic region at 96 HPF when they exposed to MPP+. So from this study, we can say that NPY plays in maintaining and differentiation of dopamine neurons during neurogenesis. So this study supports that evidence of NPY is one of the factors involved in the process of neurodegeneration. Based on the results, we may use NPY as an early diagnostic marker for dopamine neuron degeneration. So thanks.
She said this can be wide for a diagnostic purpose. She checked the specificity and the sensitivity of the test.
Okay. So you just now mentioned this gene can be can be used for the diagnostic purpose, right?
Yes, sir.
So have you checked the specificity and sensitivity?
So we are working on this, sir.
Working.
Yes, sir.
Very good.
I want to know about the zebrafish for the study of the neurons. Is there any electrophysiological study that can be done on the zebrafish here?
Okay. So in the zebrafish model, right? So is there any possibility can work on physiological
Electrophysiology.
studies?
Yes, sir. In our lab, we have done using some in project. In other project, we have done that.
But you have not done. But it is possible, right? For you.
Yes, sir. That's possible, sir.
Okay. Good. So that's it. Thank you, Mr. Smita. Divya, sorry. Divya. So can we go back to the last presenter? So you just now presented for OL8, right? OL8. Because OL10 had some issues, so we changed to OL8. So the last one, Ms. Sushmita, OL8. Can you present now? Just now we finished this OL8.
Yes, so this
And now coming is 10, but it's not around. Sushmita? It's not around. I think that's it. We have to close it. It's not around. So 10 is not around. So all presented, right? Hello? Okay, thank you very much. And you can attend our closing session through the YouTube channel which we provided for you. So to know the winners and etc. So thank you. Bye bye. Okay, thank you all for attending this session. So there are little bit hiccups in the process. So then we go for the lunch break, then come back by 2 o'clock for the next the keynote speaker. So which is our dean, Prof. Rusli Swan. So come back by 2 o'clock. Thank you. Prof. 2 is okay, or we want?
Yeah, sure.
Please go for lunch and assemble at 2 o'clock. Thank you. Yeah.
Today, this conference a lot of sitting. Oh, very tiring. Yeah. Sure, going for an hiking. It's a long.
You'll go, Prof. One weekend.
You want this place in daylight, very nice. I used to go with my wife. They have the
Happy. He'll organize profile later. Thank you. This one is actually kind of like. It was not appearing just now. Now it There also I checked. It's not appearing. Maybe that loose connection or something. This one.
By the time second day, on the third day, only president, speaker, chairman, MC.
All. Before we start, I just want to remind all, kindly ensure that all mobile phones are in silent mode or switched off. Thank you. Moving on, let's now welcome the next chairperson, Dr. Anthony Summerett, to introduce the next keynote speaker. Please welcome Dr.
The mic is there? There. Stand.
I think it's
They're holding in this. And then he gives speech.
Yeah, good afternoon, everyone. I'm really taking much pleasure to invite our prof for the talk. I must say that I'm fortunate to introduce our keynote speaker of this session, Prof. Rusli, who is our dean and the best friend for everyone. So the thing is, actually, whenever you see him, the only person who can come and speak in faculty is our prof only. He never discriminates. He is student, he is senior, he is junior and all. So he is friend to everyone. So the thing is actually we can learn how to behave to other human. So I have seen him a great human always. I always tell this to everyone. And I am very happy that I still serving under his as a deputy dean, and also I am following his steps. So thank you, Prof, for that. So I about to introduce him. I got his resume, CV I got. It was 81 pages. So I really wondered how to write and how to speak. So to cut short that, I was cutting short, it was coming 45 pages. Then again I was cutting short, it was coming 20 pages. Then I decided, okay, if I speak means I will be speaking more than Prof. Husli. So I do not want to take much time. Right? So Prof is saying something.
One suggestion from Prof.
Session is for one session for him. Yeah, I am speaking that.
Make it half page.
Make it half page, right? So I made it. I tried to make half page. Page is half, but 2 pages came. So he is the one who teaches epidemiology, public health, cell biology, whatever the science is there, he knows, and also he teaches you life lessons. Whenever you speak, he says story. That means that many story he knows, and he gives the moral of the story also. So his academic qualification MBBS he got by 1980. That year I didn't even join kindergarten. So if I might have come to Malaysia, means he may be having he may be having me on his shoulder. So unfortunately, I did not come. So then he finished his master in public health in 1984, and he finished his doctorate in medical sciences in Japan in 2001. And he got more fellowships. Again, I told you 82 pages means how many fellowships it will be there. To special mention, Royal Society for Public Health has honored him with some fellowship. And then he is a fellow of Academy of Occupational and Environmental Medicine. And more is there. Then professional member. Still now I am trying to become a professional member. I became only one, but he is 1,000. So the special mention, it is International Commissions on Occupational Health. That means he will be coming and analyze everyone and whether the system is proper or not. And he is Vice President for Asia Pacific Academy for Psychosocial Factors. So these are all special mention professional members. And he is editorial member of MedMed Central Journals and Journal of Medical Sciences and so many journals as editor, guest editor, and other positions. So he has honor and awards. To special mention, PVC Award for Excellence in Teaching. Every year he got the same PVC Award every year in Excellence in Teaching when he was teaching. And then overseas consultant for Social Medicine Promotion Foundation from Tokyo, Japan. That means international recognition he has got. Before joining MAHSA University, he served in Taylor's University as professor. So we are blessed to have him as dean here. And he has experience in more other countries, very especially Australia. And he has visited more countries. So we can list the countries what he not visited than what country he visited. Then he is reviewer of high impact journals. And he is external examiner for more universities. And his research experience can be seen by through 150 plus publications and 4,000 citations. So which is not easy for anyone to reach. This height is really very, very high. Then PhD guidance, I just wanted to number that. It was going 40 pages, so I could not number. I guess that many he has guided. I guess the number will be exceeding who are all sitting here. So that is great. So again, I'm thanking for giving me a chance to speak about him. May I now welcome Prof. Husli to deliver his talk.
Bismillahirrahmanirrahim. Assalamualaikum. Very good afternoon. Thank you, Dr. Anthony. That was a very, very touching, and I think HSC can be reduced to half a page. Yeah. Thank you very much. In the interest of time, again, I thank you for all of you still around. Because I said when we go for conference, first day full, but second day sometimes only chairman and the what do you call this, MC, and then the speaker. Yeah, we had that before. So thank you very much for keeping up. So this is the last thing. What I'm going to share today is that, first of all, before I start, again, I'd like to share with you our colleague, Dr. Sun Siu Chu, who was operated last night. I think she's stable. Alhamdulillah, I would say thank God, because she had multiple spinal injury, and they had a 9 hours, I think, operation, spinal procedure. So we still praying to God that she will recover. So still remember in our heart that we all of us pray for her. She's one of our senior colleagues in our faculty of medicine, bioscience and nursing, and always we keep her in our memory and our heart, so that we pray to God that she will be okay. Right. So on that note, because what happened to her has to do with what I'm going to say today, So that makes it more poignant, that this point about road traffic accident, injuries and deaths. It is really accident, injuries and deaths. Because when you are involved in road traffic accident, every day in Malaysia you can see, every day there's accident, every day. Normally you can have 1, 2, 3 deaths, and sometimes 3 or 4 car pile up. So it's a major problem. It's like an epidemic now, right? In not only Malaysia, I think many other countries, but Malaysia stand out terribly famous for this accident. Yeah. So today I'm going just for interest of time again, I'm going to share with you not the statistics on this one. I think that one will bore you. You can find it on the online. I'm going to inform you what are the plans now that globally, and through the WHO, United Nations, what they are planning to do, what they are encouraging countries to do. Because we can hear the statistics, the epidemiological data, but what do we do now, right? So, and today I'm going to share with you, because basically as a research institution or university, we must find a niche of what we can do to contribute, okay? There are many things that we cannot do. So today I think we will do it. So this is the latest in the world global plan. They call it the Global Plan Decade of Action for Road Safety. This is the next one. The first one was 1911 to 2020, right? And in that plan, the previous 10-year plan, there was no concerted effort to look at road safety as a system failure. In other words, the accident, the injuries, the death is a system failure. I think this is now a trend around the world, especially research, to look at this problem from this angle of a system. Why is there so many accidents? Why so many injuries? Why so many deaths? So this is what I think the purpose of this global plan for this coming decade now. Now, this year 2023, right? We already passed 2 years. So we are looking now what countries have done. And this again, we look from this perspective. So just to share with you just a small statistic from this. The number is staggering, which are these are preventable deaths, right? Preventable deaths, you see? 1.3 million. This is life. Every single one is life. Just like we have COVID deaths, right? When people look at COVID deaths, they thought this is just statistic. No, it's people. It's you and me. Remember, those who died are the dearly loved. You just imagine that it happened to us, we or our family. Then the message coming very, very clearly, right? And 50 million injuries. Can you imagine? Injuries. And these are young people, children, young people, worldwide. You can go to this one to check the statistic, or many more statistics. I don't want to share it, but we can go for the statistic. So basically now it's a killing of young people, children and young people. And then they estimate that 13 million deaths, estimated another 10 million. And 500 from this one, tenfold injuries in the next decade. So what it really means that despite saying that we are, going forward in terms of modernization, with the artificial intelligence and so many things, they're making life so easy. But sadly, we are not tackling this. Okay, not tackling this. So as I said today, like in Malaysia, we're very sad because every day you open newspaper, you don't open newspaper now. Like you just go on, you just Google and so on. You see deaths everywhere, every day, on our roads. And one of the biggest, from my perspective, because I'm also a occupational health physician, occupational health. This death we call it, or this injury I call, these are the commuting accident and injuries. I presented one paper in Tokyo to show the trend, but I won't share here. And this again is on the upward trend. Commuting accident means accident for people who are coming to work and going back to work. And that's what happened to Dr. Sun Siu Chu. She was going back to work from MAHSA. So she is under committing accident and injury, right? Injury. And of course, that is under SOCSO is compensable. Yeah. So this is again a big wake up for all of us. And this is unacceptable. The numbers are unacceptable. Nobody can accept this, right? Just like COVID as I mentioned. And has not changed for the past 20 years, despite UN or the big name, international, WHO, you name it, what they have done. It has not changed. So, therefore, now, this plan has been reorganized, okay, in collaboration with the United Nations Road Safety Collaboration, and they came up with this. Hopefully that all countries like Malaysia, which are subscribed to the WHO and to UN Charter, we have to follow suit. Okay, so let's look at the plan, this one. What do we do? What is being done now? So I mentioned earlier that this is now taken as a system approach, system failure. As I mentioned, the philosophy now is about system. So instead of system failure, so they look at the component of the system. These are the components that are listed here, multimodal transport, land use, safe road infrastructure, safe vehicle, safe road use, post-crash response. I think here people who are dealing with emergency medicine, post-hospital management, right? Really after many of the deaths I noted is because after accident, delay in transport of the victim to the hospital. Delay. Sometimes they call for ambulance. Delay. So this again something that government must do something. How to accelerate the transport. In many other countries, they use even helicopter, like drone, quickly to the scene, because every second count. People are dying. They are bleeding. They're bleeding and they're dying. And that's why some, I think Prof. Ali here is not here. Prof. Ali emergency medicine will be very, very interested in this. People who deal with disaster medicine, emergency medicine, this one. Okay. And then legal framework. We have laws. So many laws in Malaysia even. If you drive wrongly, they sue you. Okay, for paisa lah, call it. The moment they catch you, speeding limit, paisa money, right? And they say paisa nahi, mabad nahi, yeah. So again, so law, don't worry. Law, they have many law, Malaysia, right? And the police, PDRM, road transport, and so on, many law. And gender, surprisingly gender. Now they recognize that there is a difference in the risk of gender of men and women when they are on the road. There's a risk. They discovered most of the statistics suggest that men are higher risk. Is it because men are more vulnerable because of the behavior, right? Behavior. Later on, I will share you some of the study that we did in Malaysia, right? Particularly this gender difference. But I don't know whether this will stand. If any of you would like to do another gender study to look at this difference. This is positive you all, those interested in do study. My group, actually myself, I'm representing MAHSA. My group is with UPM, right? And also MIROS. MIROS is the Malaysian Institute of Road Safety. We are in the group. And we are publishing first two papers. Later I show you. So what we are doing now is we collect data. We want to actually to showcase this tragic thing about road safety in Malaysia. So bear with me, so these are the things that we are done looking at. Multimodal transport. Because we're not, as you know, many countries now, the old method of transport is gone. Now modern, right? And then in Malaysia now Tesla coming. You know Tesla? Auto driving, right? Now imagine even robot are starting to drive our car around. And Tesla, I know, not foolproof, it's not say really safe, Tesla. People have died, no? Suddenly Tesla, for some reason, something goes wrong, it goes to accident. Yeah, Tesla. So that speaks volume about Elon Musk, be careful. So this one, and then land use planning. Okay, must be also know where to site your roads and so on. Safe road infrastructure, very important. But in Malaysia, as you know, we have this highway plus and so on. They make a lot of money from this toll and so on. So they are the vanguard of safety on the road. Safe vehicle, quality vehicle. Do we have quality vehicle? Are we shortchanged by people that we bought vehicle which are not roadworthy? Okay. So these are things that now become part of the system approach now. Safe road use. Safe road use. You just imagine, very dangerous. Even you cross zebra crossing, people come very fast now. In some countries, the moment people step on the zebra crossing, nobody can go, isn't it? Stop. But here, scary, you step, people come very fast. So there's an element of unsafe in this scenario. Okay, this is again safe road use, very important, especially pedestrian. And also in Malaysia, particularly, we found that in terms of the highest number of accidents among the so-called motorcycle drivers, motorbike. Motorcycle drivers, there was a study done not so long ago, 5 years ago, that if they involve an accident, the motorcyclist, only two outcomes: major accident, major injury, or death. No moderate or minor. That is why I don't take motorcycle. My father would prohibit me from. I cannot drive motorcycle. No? I'm telling you now, I cannot. I straight away go buy car. No? I bought small car at the time. When I was in Australia, I bought the Beetle, Volkswagen, with my first car. But I never drive motorbike because it is very dangerous. But in Malaysia, so now no choice. People are using it for many things, Grab and so on, to make livelihood. So motorcycle, and furthermore, motorcycle, they go in out the traffic. You see that? You just may all of you driving, they without any signal, they easily looking for trouble. No? In many countries in the West, these are dedicated lane. The major highways, no motorcycle can go on that lane. They're only on the side. But Malaysia, unfortunately, they don't easily go. So this is why they come. And then many again who become like this what do you call it? Hero, like Spider-Man on the motorbike, isn't it? Superman. What call this? What call it? Mak Rempit, So this again safe by vehicle, okay? Safe road use, and then safe road use, and then post crash response, okay? But what was not glaringly was not shown here, in the way maybe here, it's about the behavior of drivers. Behavior, and this is what our focus now in our research is the behavior of drivers, especially when it comes to using phone, texting, okay, eating. Sometimes I ask people, they say they feel the moment they go to the car, they become hungry. Correct or not? You don't mind. You even ask yourself, at home you don't feel hungry, The moment your father start the car, every day go in, then suddenly you get hungry, Then all the food finish, Somehow there's that. I think there's the effect of going on the car, because driving, then you feel happy, start eating. So eating and drinking. But if you eat drinking, okay, but you drink the wrong thing, drink alcohol and driving, or taking drugs. In Malaysia now, commonly these are long distance drivers, a bus, a bus and so on. The driver, because they are taking trips to make more paisa, taking trips. Therefore, they are cutting them because they're not sleeping actually. They are taking methamphetamine. I'm telling you this is all study, but I'm not disclosed here, right? So these are some of the behavior that we found that can cause many of these accidents. And the one that Dr. Sun was involved, I do not know what's the cause. It could be easily that the person is sleeping at the what do you call this steering wheel, what we call now is a micro sleep. There's no problem now. It's an issue about micro sleeping at the wheel. Few seconds of sleep, you are already accident. Right? 1 second, because you are driving, at speed. 1 second, you close your eye, is boom. Okay, so micro sleep. So this somebody is doing study now on that. Our group also looking at micro sleep. Okay, and the other one I already told you, this thing about craziness about this addiction to a smartphone. All right, you're texting and so on. Even listening to music, Spotify, phone, all these things are distracting you. So these are things that we have to look at. And then legal, as I said, it's not too much. But this gender is a special thing. Gender, I think we also would like to do another study to look what is it that makes the risk of accident, and also and death, injury and death is so different across this divide. Or are we wrong now? The hypothesis can be, you can doubt the hypothesis. Actually there's no difference. Because like in Saudi Arabia now, for instance, example, Saudi Arabia. Many years ago, the ladies cannot drive Saudi Arabia. Not because they don't not allowed to drive because of Islam. No. Actually, I asked the fellow, why you didn't allow? Because they say in Saudi Arabia, if the woman drive, if the car break down, there's nobody around, she may disappear. Saudi Arabia, if you disappear in the desert, nobody knows. It's not a safe place, Be careful, So now I think because they discover that it's becoming safer, so women are not start to drive. So maybe that will equalize it. Huh? So that's the gender side. So what do we do now? Okay, the thing is that it says that we declare that the National General Assembly Resolution 74299 declare of action with the target to reduce road traffic deaths and injuries by at least 50% during this period, this 10 years period. By 2030, the figures that we have now should be half. At least should be half. If we are actually diligently following this, and all countries that subscribe to UN, including Malaysia, subscribe to this and follow, right? And what do we do? So many things here. As I said earlier, now the issue that we're looking at it from the system angle, system failure. So system. What system? First, paisa, financing. Only paisa. This one, if without money, forget it, right? This is the main driver of this change. And then government, politics. Who is in charge of government? Do they prioritize road safety or not? And government are very sensitive to this because why? If they don't, they may lose the next election. Okay, example. Yeah. And then speed management. What is speed limit in Malaysia? Anyone knows?
110.
110. How many plus drive below 110? Even I myself drive faster, no? Because if I drive below 110, I get very sleepy, So some stretch I go very high, so very bad for me as well. I give lecture today, but I myself drive very fast. So I need to also control my speed. So speed limit, because it is true. You just check on the internet. Any of the study have confirmed that the higher the speed, the higher is the risk of accident, injury, and death. That is very clear. This what happened to soon. Because the car that hit her from behind. She was driving a Maybach, The Maybach was crushed. You just imagine, if somebody is at the back there, occupant, instant death. She was in front. She was caught, and then even the airbag could not support her during that crash. Can you imagine? This airbag are not really safe actually. I mean, they say not safe. It cannot totally protect you. So today, one research they do now that they have produced a jacket that act like this airbag. The moment accident, boom, the jacket goes up. I think I'm buying one. But this jacket is meant for motorcycle rider, not for car. But it's very good, no? They try. The moment it's accident, boom, come on. You become like astronaut, no? Like that. So I like that. So that's one innovation and one research done because under this decade, to find out how exactly can we reduce the accident, injury, and death. Okay, then capacity development, manpower, critical. Who are Malaysia? What do we have? We have only police on the road sometimes. Plus, okay, who else? Not many. In the city town we have from this local council, and then the rest maybe NGO and so on. Civil society here, academia at the top here. Academia, NGO, youth. Right. So you imagine, because civil society are very important. Private sector, technologies here again, AI. We talk about AI, more and more. Tesla car. The other day we had a talk by one guy, going to space. The one of the talk is that Elon Musk is going to go to Mars to relocate. So the Tesla is just one of the experiment on Earth to have a car in Mars. Tesla like. Don't know how they're going to do it. Okay. Then focus, and then the focus on low and middle income countries because still this problem is very high in low and middle income countries for obvious reason. Yeah, very low infrastructure, less paisa, and so many things. Yeah, so always this one. If you go to Western country, because like Germany, I've been there. Germany, although you have the autobahn, very fast, but very rare accident. I don't know whether I have ridden on the autobahn in Germany. There's no speed limit. Yeah, autobahn has no speed limit. You can drive, but surprisingly, not much accident. Something like in Indonesia when I go there, no many accident because always jam. They call it macet. Macet means jam, so they cannot go fast. So no accident. So you imagine, I went to Thailand again. What if you accident in Thailand? I went to Vietnam. The system was very simple, but no accident. I was wondering why Malaysia so many accident. In Vietnam, a lot of tolerance. If you are driving motorbike, no? You are a car. If you hit the guy hit you, you are at fault. Just Thailand. The principle that in Thailand and Vietnam is that the bigger vehicle is at fault. So just imagine if you are cycling, it's very safe. Nobody there to hit you, no? Correct? Yeah. All right. So that's funders are critical, but it's not easy. Funders who can fund this program, and then of course these are more of monitoring. The UN must remember, no, they are more of monitoring, giving you information, no, statistics and so on. But they're good in terms of charting where we are compared to other countries. But that's what they're but ours is here. We are academia. This is where research comes in. So therefore, here it says that road safety is at a crossroad. Crossroad because in the previous decade, it is the principle is business as usual. They just do what they can. No country come up with their own plan, no? Business as usual. But today we have already another part of the road is that system approach, integrated system approach. We have to look at this from the system angle. I think that's the way to go. All right, so therefore there is hope. There is hope now. Okay, and making safety a core value. That's why safety now, even Malaysia now become a top one. If you have any experience of OSH, occupational safety and health, which I'm in that, you find that it's coming bigger and bigger now because safety is very big issue now in Malaysia, right? Because of our so-called unsafe situation here. So just to get it from this angle, okay, it's a vision here, right? This is a vision here. So the vision here for the next for current 10 years, we have to look at all this from the safe system. So if you do a research, you are contributing from the system angle. Okay, you're providing information from the system angle. All right. So these are some of the things just to show. Okay. So these are the things. I'm not going to bore you, but just simply say that anticipate, accommodate human error. So human error is the biggest one now. All right. And then road and vehicles design, motivation to design road, maintaining road, manufacture vehicle safety, responsibility. So many, commitment to proactive continuous improvement, and then to improve transport system. So it is depending on what's your interest. Some may be interested in the engineering part, maybe design of vehicle. It's up to you. Design of safe system. So you can actually accommodate many of these features. Okay, so that's why we come back to the recommendation. I just keep many things here, but I think great, multimodal transport and land use planning, very important. Okay, in many countries, they are now going towards more the greening of the environment, less pollution. All right. So what do? Encouraging more bicycle. But of course, if you don't have bicycle, you go 100 kilometers, you can go by bicycle. But encouragement. In Malaysia, difficult to see, Have you seen any professor coming to work on the bicycle? I should have bicycle also because our roads are not safe. We do not have a dedicated lane. In many Western countries, dedicated for motorbike, for bicycle. You cannot hit them there. For car, separate. But here, everything in one, you can get many accidents. So this one, costly but must do. Secondly, when I was working and studying in Japan, in Tokyo, Japan. So this Japan. So in Japan, they have a lot of using of public transport, subway, train. Expensive, but they do it. Subway in Japan. So Prosuba also work in Japan under GSPS, also under GSPS, PSD, this Rompaku, So there, the system fantastic, Nobody can tour with their own vehicle. Everybody go by public transport. In Malaysia, you try public transport. You say to take a bus by 8, they come at 9. All right. In Tokyo, you go subway, every 5 minutes, train coming. Every 5 minutes. You want to go far from Tokyo, I go to Osaka, Shinkansen, the bullet train. Okay, very fast. So just imagine that so-called system are so different, no? All right. So this is again multimodal transport. No? Basically, what I think Malaysia now the very important to upgrade is so-called public transportation. Railway, we have MRT, no enough. Underground railway. So on the surface, underground, we must have. The next government, If I know how to stay, he must have this thing. All right. Overground, above ground, underground. And paisa, of course, again, money must be there. You must allocate big money. Okay. And then the rest, like buses. I stayed in Melbourne 7 years, Australia. Right. We have trams. Trams, imagine, nice, trams. When I was a student, I worked as a tram conductor, tram driver also. So I'm talking from experience, not just I'm going as a passenger. I am a conductor, I'm a driver when I was medical student. Imagine? So from experience, fantastic system. All right, so this I think, again, not to bore you again, so many things, policy and so on. A lot of policy. But the things that these are things they can recommend from UN. But can we do it? My fear is that one of them also is difficult to do. So let's not be worried about too much long list of things. All right. Secondly is safe road infrastructure. Again, very important. Maintenance road maintenance. But Malaysia, I think not so bad now. Some countries even worse. Pothole, no. I went to Indonesia one time. I went to Kerinci. Pothole, no, big pothole. I was so scared how the taxi I took are able to negotiate the pothole. No? I don't know. I salute the guy. If I already inside the pothole. Can you imagine? Right? Okay. Safety again. Safety. This again about having with Malaysia we have two is our own proud standard Proton, but now already occupied by Geely from China, and then we have the Perodua, right? And then the rest, as you know all from Japan. Okay. Now China is making a lot of car to Malaysia, good quality car also. So a lot of competition there. Right? And then of course the European car, your BMW, and then your Mercedes and so on. Yeah. All right. And then again, safe use. All right. Legislation we have that. Licensing we have that. We have that. Vacancy feature we have that. But again, how much? Some are talking about safety belt and safety seat. Okay, speed assistant technology, AI. And then post crash response, so important, critical, post crash. After you have accident, okay, system to activate post crash response capacity among lay responders, strengthen professional medical care, establish requirement multidisciplinary post crash investigation, social, judicial, and financial. So many things. But to me, critical now is that to provide the infrastructure and also the speediness of to the crash site. As I told you, many crashes where people die and injury become worse because of delay in providing assistance at the site of the crash. And also sometimes I notice if people are not well trained, even carrying the patient could aggravate the condition. If you have neck injury, I am also a first aid trainer, right? I train when I was USM. I'm a first aid trainer. You cannot simply carry people, especially with a sensitive fracture like neck fracture as well. If you don't know how to transport, this will come under transportation. You will aggravate the fracture. That's why if you don't know, leave the patient there, unless you're worried that the vehicle is going to explode and so on. Okay, you leave it there. Let the professional carry. So this is again another one. Sometimes we thought, what come this person sustain severe? Then we realize that it's not just from the accident, from the failure to transport properly the patient. This is the thing, right? All right. So therefore, in this case, we are going to summarize it. Yeah. These are the 7 points: financing, legal framework, speed, capacity, gender, developing technology. So it's up to you all. I know you all are doing research mainly in bioscience or even biotech, bacteriology. But if you are interested in this research, you can even call me because we have this group. I said now we are looking at many of this area. We are developing a questionnaire. Later, I can even share with you a questionnaire looking at the behavior of drivers. We are this about second paper we are submitting, right? So shared responsibility here. All right, we mentioned again and again. Shared responsibility, monitoring, evaluation, very important. Is under UN. Okay, and the way forward, as I said here, in our midst, what we can do, not only us as in academia, what we can do? We do research. All of us do research. Yeah. Yesterday you heard from Prof. Subeke, Prof. Subhas, Prof. Bob today, all about research, lab and so on. So the thing is that they came here is about, can we do research on this? My area now we're focusing on driving behavior, because we found that more and more that the culprit, the causes, the root cause of accident is behavior. Sleeping in the wheel, why? Because you are too tired. You are supposed to rest at the R&R, supposed to rest. Whenever you're tired, always the advice that you must rest, you go there and sleep. And sometimes when they sleep at the R&R, they die also. You know why? Carbon monoxide poisoning. Because when you arrive at R&R, the switch is on, you up all the window, right? If your system, your combustion is not good, many of these carbon monoxide, which is odorless, colorless, you cannot smell it, cannot see it. It go into the car. Recently, meaning about 1 week ago, 3 people from Myanmar died at the because how they discovered, because the car was there, it was too long, the engine was too long. People got suspicious, it was so quiet. They knock on it, no response. Then they smash the window. They found the 3 unconscious, died. Then they declared, go to hospital, check, carbon monoxide poisoning. You know how it displays the oxygen from the hemoglobin, isn't it? So this is very dangerous now. So if your car is fault, come back to the same design. Car is faulty, your combustion, you have to rectify. Otherwise, you get this thing, you die from carbon monoxide rather than actual accident. All right, so just to share again, this example, the one we are developing a study protocol, prevalent predictors of road crash involvement among medical doctors in Malaysia. Why this study we did? This is my group from MIROS and UPM, because the last 1 year, we heard many doctors died, especially housemen MO, because when they are working, they are so tired, because imagine they are working, sometimes they are on call 24 hours. So by the time they go back home, they are very tired. Many of them died of accident because they're sleeping at the wheel. So we asked around, anybody has any questionnaire? Then we discovered that nobody has designed a proper questionnaire. So what we did now, in this study protocol, we designed a questionnaire. The questionnaire is second paper. We already submitted for publication. So if that questionnaire is approved, then I will let you use it. You want to use it for your study, all right? So this is an example and the fact here, see, traveling to and from work. Before morning shift. Why morning shift? Because you're rushing. Rushing because you're scared that you will late to clock in. Especially MAHSA. After some you see don't have clock in, clock out. MAHSA is clock in, clock out. So rushing, you look at the work, 7:30 already, I'm rushing. I must reach before 8. So this is why we found out that before morning shift. Not after work. After work, Leslie, director, you're going out, drive carefully, put one Spotify song, Santana or something, you can drive, okay. All right, sorry. So this, and within 5 kilometers of travel distance. And motorcycle riders having the worst casualty, motorcycle. This one is already based on study. We know. So this again area that you want to research, you can do it. Look at this so-called behavior of drivers. How certain behavior actually increase the risk of accident, injuries, and fatality. See, these are some of study from overseas. Study 70 junior doctor revealed that 40% had fallen asleep when stopped at traffic light. Can you imagine? Traffic light, you arrive, red, stop, and then quiet, and then green, stay quiet. Then we are sleeping. Sometimes we scared they get heart attack. So quiet, sometimes I knock on the door, I'm so scared, I thought heart attack, Oh, holy. And then actually sleeping, too tired, yeah. Sleeping, okay, but heart attack, another story, right? And 49% had fallen asleep at the wheel. At the wheel, Traffic light at the wheel. What's the difference? Different. Traffic light at least something. You are still stationary. But at the wheel, you are driving. Oh, finish. Finish for you, yeah? 90% of this event occurred after long working hours, post-call. And 20% of them were involved in road accidents and injury. road crash involvement, RCI. So again, some references to show you. I think that's it. I don't want to, again, I know it's very long already for me. These are the things I want to share. Just to conclude, basically, I think from our angle, what we can do, I think we should actually think about it seriously, as a very fertile area for research, especially a safety feature, behavior part, mental health, use of substance during traveling, like what people are still driving under alcohol, using methamphetamine, all these things. All right, stress. Marking about Malaysian driver, they say, when they leave the vehicle, they are like angel. Malaysian drivers, when they are not driving, they are angel. The moment they get behind the wheel, they become devil. You know, suddenly they transform, from angel to devil. Very scared, So something in the driving behavior changed the guy. I don't know why. I hope it's only now the study only among men. I hope it won't have a female devil behind the wheel. All right. So this again thing that we need to study. How can people change when they go behind the wheel? It's just something about the aura of driving, giving them that feeling of grandness and so on, and recklessness. The word reckless, because you just don't care, whether you are injured, somebody injured. Right. So on that note, thank you very much, ladies and gentlemen. I hope that this give you some idea about what research can do. Thank you. Sorry, is there any QA? Anyone want to ask? I forgot what QA. Sorry, it's none on bioscience and biotech, but on safety, on road safety. Anybody interested to do research on safety, behavior, gender different? Yeah.
Yes, discussion. Yeah. Previously, means I used to drive in India. I never used to follow signals. Very less signals.
Oh, really?
Means less safety. After coming here, I whenever we have to take the left turn, I used to give 30-second signals like that, and I used to cross the lanes and all. But now here I observe that they are not following the rules.
I think. So why?
Don't.
This is what I say.
Thank you very much. That's a very good observation. All right. You just remember, it's not. I mean, we cannot just say, "Oh, we catch him and then find him." The one is paisa, but this is about behavior. Behavior. Why? See, all of us go through a driving license, isn't it? Before we get our license, we go through a driving class. All of us. My driving class was in Melbourne that time. Although my teacher was not teaching me well, he said something else. Anyway, I passed also. So, but driving class, you go for classes, you pass, you got your driving license. But as you said, why are they not following? They thought, they're important. All these things are taught. So this again, to me, research. Why are people? Is it the current generation are purely careless about safety? I don't know. Is it?
Prof, as you said, this is one of the major reason that now generation is not following that.
Exactly.
You don't care for the.
I don't know whether it's a generational issue, rather than what. Not to say that the previous generation, like myself, only worry about this safety feature. I don't know. That again, research. Then we must find out why. Is it the current generation they are purely careless, because they only playing game, so many game, simulation game, virtual game, okay. All right, augmented reality, virtual reality, extreme reality, all these things. I don't know.
Okay. Thank you for nice information. You showed so many the data, something like micro sleep can be. But the difference is people are using a mobile phone. That should be the high.
Yeah, correct.
A high percentage for accidents.
Yes. But surprisingly, because why? You see, the things that many of these captured, maybe people are not telling the truth. Actually, the real thing is the microphone. I mean, the mobile phone, isn't it? See, the more we just have discussed yesterday,
Yes.
It's an addiction now. We live in the decade of mobile phone addiction. You like it or not? No, it's an addiction. And not only that, robotic world. we always talking this day, one day you also will marry to a robot, no? You know, no joke aside. In Japan, one fellow married a robot, no? Robot's wife. They went to them, the usual Japanese marriage. I love you, you love me, and that's kissing, happen. But married to a robot. Then when they get baby, maybe half man, half robot. I don't know. Alright. So okay. So this is where I think your Professor is right. We know that smartphone, but why it's not? Is it because they are not telling the truth, no? In the survey, or in the study? It should be higher. Higher. Must do research, isn't it? That's what research is. Find out the truth. Maybe you can do some focus group, some qualitative. Ask them quietly, why didn't say the smartphone? So this is important, not to say we ban smartphone, but I think there should be something to do that some moratorium. I saw yesterday somebody saying while driving they could have a system that deactivates smartphone. We cannot use. You more like shut off the internet from there. So we can do anything with the smartphone except for maybe calling or play some music, satana. Example, deactivate. The moment you go to deactivate. So that's very good. Now this research should be shared.
As you said, Prof, like just the last month only, as I told you, I was working on the social media addiction globally for latest stats 2023. So from the stats that I got worldwide, almost 70% of the drivers were driving the use social media. So that's the major cause for the accidents also, negligence actually.
Exactly.
So they are busy in that. And I compared the Malaysian stats with the worldwide stats. In Malaysia, it's even higher.
You're right. Yeah. That could be the fault, right? Now, I mean, we have to be honest and accept that all of us are using the phone in the car in one way or another. Sometimes, guilty, you put there and suddenly you want to call somebody. As you look at the phone, you're distracted, isn't it? Every second, is accident, especially when you're driving above 110. Every second is an accident. You turn your eyes there, accident. Even that day I was driving Chandra, one motorbike we saw in the rear mirror, we only got a blind spot, If we turn, the guy already crosses. See, we don't know when he crossed. Suddenly. That's why I said accident is a split second, and it's not minor. Motorbike, I told you the study research shown is only major or death. No motorbike will sustain minor or moderate injury. Major injury or death. That is what, and all death. So this is how bad. And motorcar, same thing. The strong motorcar in the world that I know, I used to have old car, Volvo. Volvo is a Swedish car. If you want a car, you don't mind paying a lot of money because paisa, because the petrol, take Volvo. Very strong car, very strong.
One of the safest. This is the safest.
Safest car. It's like a tank, very heavy. If somebody hit you, you don't even feel, You say, the fellow, and you see at the back, the car smash, But you're okay, It's very safe, but very uneconomical. Yeah. So that's an example of car design for safety. Because the Swedish, they are more on safety, but the Japanese more on the economy. There's a last time one car during my time, you're not born yet. That's a sunny. It's like you call it a moving coffin. If accident means you're dead. Even you go and you punch the car, you'll be dead there. Can you imagine? You can even push it there, there's a den. That's how cheap the car is. And that's how cheap your life if you drive the car. It's called Datsun Sunny. That's it. Any other question? I have to stop.
Same as you said, Prof, like micro sleeping. So when we talk, I can even feel like microseconds of action. So microseconds only. So we should also look at these reports as well. Thank you, Prof.
They're using some sensors.
Yeah.
That anybody sleeping alarm.
Correct. Yeah. Maybe you can design some sensor. Yeah, correct. To trigger the alarm. Today, Dr. Mo using some biosensor, on the arm. Example, similar thing, a gadget, that will trigger you, or maybe even your watch, it will trigger you. Maybe give you some pain shock or something that you are sleeping, I don't know. These are research. Please do it. Because if you do that, you are doing a good service to humanity. Actually, you do. You will easily, get the below 50% target. Any other question?
Just to share
Yeah.
the latest cars.
Like
I think in India also it is there.
Latest car from Portugal. Like when I'm driving, if it is sense,
Alza.
It offers me tea, coffee.
Really?
Like a picture.
Oh,
so it notices the way of driving. Virtually, it looks at me the way I drive. Honda CR-V and Alza, they offer in the screen, screen, and they will show me a cup of tea. I think with the message also, I think you need to have a cup of tea break.
It's an indication for you that you just wake up.
Yeah, wake up. That you're not driving according to what your usual pattern is.
I want to go one on that note, Dr. Ivan, I want to go one more. Maybe one day we should have a robot in the car, no? To slap us. The moment you are out, then you wake up. What do you think?
Even better.
Yeah. Maybe we can suggest a robotic aid.
When we are sleeping properly, even the alarm will not wake up.
Yeah.
So a slap is good.
We are driving properly.
Yeah, really. You're right, no? Stop slap, huh? Anyway, on that note, no question from student. Nobody want to ask question. Don't be shy, no? Very interesting. Not interesting. It's a serious issue, actually, very serious. This death, this how we call it avoidable death, preventable death. You can prevent it, actually. But unfortunately, people die. No? Imagine now, we have not gone through the effect of the family in it. How desperate they go to what you call it, depression and so on. Then paisa again, no. The breadwinner, we say the father passed away, no. Who to take care of the family and children? Huge implication on the family and so on, on the nation. All right. On that note, thank you again very much, and I hope that all of you have enjoyed the conference, and look forward to the next conference next year. Thank you very much.
Thank you, Dr. Anthony Sommer and Professor Dr. Rusli Benorde. Next, I would like to invite Associate Professor Dr. Zenkata Suresh to give an overview of the MindMed 2023 conference and announce the winner for our oral and poster presentation. Please welcome Prof.
So all the delegates and participants, a very good evening, and we are coming to the end of the program. So we have started 27th of July with the pre-conference workshop, where around 34 participants participated by Prof. Ali and group successfully done the workshop on CPR, first aid, and etc. And after that, yesterday, that is the first day of the conference, we had around 175 registrations from 5 different countries. And it was started with the welcome address by Prof. Rosli, then followed by the opening remarks with the Prof. Datu Ikram and Prof. Rosna. Then we had 4 keynote speakers. All are excellent and very informative. I think everyone enjoyed us. And the last one, we had much more extra. We had a discussion which everyone interacted. It was very nice. And first keynote by Professor Bikosh Mehdi, where he gave a speech on preclinical and clinical study consideration for IND applications in drug development process. So it was a very informative session, and with a very excellent the examples he gave, and in order to make it more interesting, and we got a lot of knowledge out of it. And after that, keynote speaker 2, Professor Dr. Subhash Gobinath, and biomarker imprinting on integrated electrode for diagnosing human blood clotting defect. It was very informative, very motivative, and a lot of figures. And it was started from the scratch. You should see, nothing was there. Just aptamer designing until the device is a full study. Usually it takes 1 decade normally, but they have done within few years. It was very motivative, and it is eye-opening for in many areas how we can publish things. And The third one is by Dr. Mok Pui Ling, the stem cells, research advancements in and the potential applications. This is another, the regenerative medicine area research ongoing, very promising area, and she covered all the fields of the stem cell research and ongoing, and what are the potential applications of the effect in medicine. And the fourth one, Prof. Rosalie, it is road traffic accidents, injuries, and deaths, and global health, global public health issues and responses, which is a very timely talk and very important for every one of us. Not only the medicine, every human, every citizen need to know all those things, and updated information. It was shown, and very, we had participated, all of us. Discussion is very lively. So apart from that, so that covered all the four keynotes, covered all the different aspects of the medicine, and it is a fulfillment of our the conference. Apart from that, we had oral sessions and poster sessions, and the poster sessions yesterday, the afternoon session, we had 15 posters, and then we had oral presentation around 40. Out of 40, 13 were face-to-face. Yesterday was held in two different sessions, morning and afternoon. And today morning we had online session 23. So total together we had around 40 oral presentations. And that also cover different fields of medicine. And I hope all of you enjoyed those oral presentation and poster presentations. And this is the summary of our conference, a pre-conference, free pre-conference workshop, and a 2 days conference. And we had, definitely you agree with us, with me, that we gained a lot of knowledge in the different fields of the medicine. And with this knowledge, we can take away and motivate and try to do some research in future, which is needed for the society. Okay, so now we'll come to the session announcing the winners. And winners, we are providing the e-certificate, so that we are not asking them to come to the stage. We just announce and display the certificate on the screen. Okay, so before that, I would like to ask all of you to give a round of applause to all our keynote speakers. A round of applause to our oral and poster presentations. So maybe everyone might have done well, but we, based on the different aspects of the presentation and outputs, everything taken in consideration, judges made the judgment. First, I would like to announce the poster awards. The poster, first one will be first, and second is second. And we have given selected three best posters. So the first one is Miss Vigneshwari Gunasegaran. The poster number 9, cloning and overexpression of the RNA chapter 1, HFQ protein from Sedation medicines. So this is the first one, Vigneshwari, poster 1. And the second prize goes to poster number 6, effect of muscle music on cardiovascular system function. So there are many members for this research. I'm proud of you, our biology students. Fatima Shioza, Ling Chunwa, Manigantan, Maimin, Muhammad Faris, Mursalin Islam, Nick Amar Farhan, Puteri Nur Farizara, Roshna Fatima, Sara Akila, Afnan Shihab. So these are the winners. And then the third prize goes to poster number 8. Bacterial contamination on different time usage of face masks among preclinical undergraduate medical students. Very good. This is having a potential of commercialization also, as Professor Slay said last time. And the winners are These are the one. Okay. So Wan Nur Aida, Lu Yong Kit, T Wei Zong, Siti Aisha, Husna Johna Iman, Li Li, Lim Ilin, Sydney, Gunavardhana, and Godella Patirage, then Parkia Raja, and Hechi Arachige. So pardon me, pardon me if I have spelled wrong. Okay. Then goes to the oral. So oral, we had 4 different sessions. Yesterday morning, one face-to-face, afternoon, one face-to-face. Today, we had 2 oral online sessions. So altogether, each session we decided to give 2. So we have oral face-to-face. We have 3 best oral presenters. There is no 1, 2, 3, all best. So the one Professor Dr. Betsy. Okay, Professor Dr. Betsy. Miss Leongyi Ling, Miss Surya. Okay, so and come to the online as they presented many, around 27. So there are 6 members we have selected among the best. They are Miss Atishta, that is computational analysis of bioactive compounds from Memordica charantia. So then next to Miss Abhirami Saniraj, elucidation of the role of non-coding RNAs in regulation of virulence in the biofilm Fimbriae and flagella of Proteus mirabilis. And then next to Miss Maria Banu Sri Rajashekaran. Then next one goes to Miss Joy Vinushia. Then Miss Samhita Gangula. Then next, Miss Sumita Samuga. So these are the oral best presentations. So with this, I would like to end the session. I will pass to the MC.
Thank you, Professor Suresh, and congratulations to all the winners. Ladies and gentlemen, it is an honor to invite Professor Dr. Rusli Renordin, Dean of Faculty of Medicine of MAHSA University, to present the souvenir to our keynote speaker. First, I would like to invite Professor Dr. Bikash Medi.
Thank you.
Thank you.
Followed by Professor Dr. Subhash Gopinath.
Thank you, bro. Thank you. Thank you.
Followed by Associate Professor Dr. Mok Pui Ling. Next, I would like to invite Professor Suresh to hand over the souvenir to Professor Dr. Rusli Benorde.
Thank you.
I would like to request all of you to come to the stage with souvenir for photograph session. Can all the keynote speakers come to the stage? I will also request Dr. Ivan and Professor Suresh to come to the stage.
That's all. Okay, all right. Okay. Yeah.
Or you can come this way.
Okay, yes, ready?
Okay, okay.
Ready? Thank you.
Thank you. Thank you. Okay,
I would like to make an announcement to all poster presenters. Please take back your poster. We have now reached to our final part of our conference. For this, I would like to welcome Dr. Anthony Samberg to give the closing speech. Please welcome Dr. Anthony.
Thanks, MC. I was wondering will you call me or not, so I closed all the things. So you are going to close the session. So thanks for giving me an opportunity. So thanks everyone. It's really honor for delivering vote of thanks on this day. So to thank, very first I want to thank God the Almighty for giving strength and all the support for conducting this conference. I thank our management, our Dhansri, then our Dato GMD, then Dato GED for the great support. I thank our Chief Operating Officer, Prof. Allah. He is the one who really made this conference going in a proper way and everything. He was monitoring and he did a great support. I'm grateful to our Dato VC, Prof. Vikram, for coming here and for delivering the inaugural address. My special mention thanks to Prof. Rosna, DVC Research, for delivering special remarks. I must thank all our keynote speakers. They came all over here in the busy schedule. They are really, really very busy. Whenever you call them, you cannot reach them easily. So to reach them, we have to at least call them constantly. That much busy they are. They are truly busy. And they accepted and they came here without any hesitation, and they delivered the talk because he came from very long distance from Unimap, and he is delivering. That is Prof. Subhash. Then today, Dr. Mark, not well at all, but she came only for delivering the talk. That you are all very fortunate to see them. And Prof. Because he is a top scientist all over the world, and he came. And Prof. Usli, he came and he supported us. Always he supports us like something. So today he came and he delivered the talk. I believe all the talks are really eye-opening, and it started us to think about in new research thoughts, and also we feel that we'll be collaborating with them and we'll be taking the research field to the sky. Hope we'll be meeting again. In future events, hope you will be coming like this and you will be helping us and you will be delivering more and more talks for us. Now, I thank our Dean, Prof. Rusli, for the support and the guidance and the freedom what he is giving every time. He believes in each person's ability and really gives room to every person to move and excel in their way. Thanks, Prof, for supporting us, and hope you will be blessed us like this. I thank Dr. Ivan Ronald, Deputy Dean of Faculty of Medicine. He is the right person. I should say that he never asks for any formality or something. I just go to him. I say, "Sir, I want this." Yeah, we do. I want this. He never says no, and he always supported me, and he never questioned me. Also, why are you doing like that? So that is a real support, sir. Keep supporting me without asking any question. So that is very, very essential. Okay. Okay. So then I thank organizing committee members, Dr. Bharani, Dr. Reshma. Today they are not here. Don't think that they are not doing anything. They are typing your certificates. Right? So spelling mistake should not be there, and it has to be made into a proper thing. So they are working on that, still working. So you'll be getting your certificates soon in your mail. Don't worry. It is e-certificate we are giving. We are going paperless. So we are not printing. We are giving you the certificate. It will reach your mail. You can get it soon. It will not take more than a week time for sure. Then Dr. Shalini, so who is the real support, and with patience she did. The previous day also we added paper. When we went to her and I asked her, please add. Yes, doctor, I can. She was so busy with other work also, and she supported like something. And like a friend, she was supporting. This is a great thing. I thank Dr. Shalini. Right. So next time, please add my more papers, right? My papers more, and try to support me more. Thank you, doctor, for that. Then I must thank Dr. Suresh and Dr. Safi, the co-chair of me. Right. Dr. Suresh and Dr. Safi really supported me at every stand. So Dr. Suresh, normally, whenever you go and see him, he greets you with one smile. The trademark smile he is having, now also he is doing. It gives me energy. So keep smiling always, right? So thank you. Dr. Safi is busy with some other work for this only, because the posters and everything he made, so he went to pay the bill for it. So he's not here. Then I must mention about Dr. Menaga, Dr. Rajeshwari, and Dr. Rose Fayati. Excellent work. Excellent work. Because the previous day, I was thinking whether the arrangements are correct or wrong. No one, they are really superwoman, I guess so. So did everything. They said, yesterday also I asked, is there everything ready? Tomorrow morning, 8 o'clock it will be ready. When I came here, it was ready. So it is not possible. Whenever I was asking anything, they were giving it immediately. So I guess continuously I can conduct 100 conferences with their help. Thank you, Dr. Rajeshwari and everyone. Right? Rose Fayati is here. Dr. Rose Fayati is here, I guess so. Okay. Then Dr. Zain, Dr. Rice, then Dr. Chandra, they supported us logistic. Everyone knows about Dr. Chandra. He is a man supports everything. If I ask, yes sir, I'll go. Yes sir, I'll go. He never said I will not go. So I'm using this vehicle as trap now. So even night 11 o'clock, he is going and picking someone, dropping them, then taking them for lunch. So he treats everyone. If anyone wants lunch, you can approach him. He gives free lunch, no worries. Then coming to Dr. Sudha, Dr. Madhu, then Dr. Sabha, Dr. Nancy, they are all really supported. The previous day they made everything ready, your ID card, everything they made in one day. That is really a miracle. Keep doing. Then I would like to special mention thanks to Miss Hasrinda who made that tea and every arrangements there. Then Miss Elena supported everywhere. Miss Ashiara supported everywhere. Then I would like to thank Dr. Chin, Dr. Mock, Dr. Bilkis, and all the other members. I might have missed some names. Dr. Nala also, she helped us as a chair. Thank you so much, Dr. Nala, Dr. Sabha. It is really a great help. Then I thank the management of Vivekananda College of Arts and Science for Women from Trichangot for co-organizing. Thanks, ma'am, and having come here. You brought your great energy to Malaysia. So thanks, ma'am. I thank Dr. Nagaraja for bringing them here. So you made it possible. Thanks, sir. Hope you will be bringing more universities and colleges here for our future events. I thank all the chairpersons, especially from India. Dr. Maslamani, my friend. Prof. Renita, she is also my fellow colleague earlier. And then Dr. Purni for helping us without any hesitation. I went and asked Dr. Purni, please, can you please judge that? Yeah, immediately I go there. But the problem was she was going and speaking with everyone for 1 hour. So how we are getting that energy, I really wonder. So I told our IT department and FAST, switch on the light for the entire day, entire night, because Dr. Purni is on action. So thanks, ma'am, for that. Okay, so I come to the major part of thanking the persons. Actually, this is participants of our own MAHSA University, right? So you really made that. Thanks, students. And I thank students from Satyabama for your energetic support. Okay. So yesterday I asked you how the conference is going. Everyone said very fine. So it was good. But I asked about some topics. They said that time only I slept. But anyway, it happens. So when more energy is there, sometimes our battery gets down. So it happens. Thanks for that. But being inside a hall is very hard. But you made it, and thanks for that. I'm grateful to IT department and FAST of MU. They only made everything ready. So that is a real thing. Okay, if I go like that, right? At some time you may be getting irritated. So I am going to stop here because my watch is also alarming me. Don't speak more. So I might have missed thanking someone only because of no time in my watch. I have more in my mind and brain, but stopping here. So I thank everyone who supported and made this event a great and grand success. Terima kasih semua. Thank you for that.
Thank you, Dr. Anthony, for the very warm closing speech. On behalf of MAHSA University, I would like to extend my gratitude to all the professors, associate professors, invited speakers, ladies and gentlemen, for being with us here today. I hope you had a fruitful session and gained important insight from our MAHSA International Medical Technology Conference 2023. I'm Jididiana as the MC of MyMed 2023, signing off. Thank you.
We are going to take group from here. You cannot come forward. Please come forward and occupy this.
Please come forward.
Yang baru. Yang baru.
Come, come. Don't stand at the. Can you fill up the center one? Please come. No, you check and
No, cancel. No issues.
come. Those who are sitting at the back, can you come and fill up in front? Move fast, don't waste time.
Cancel. No issues.
Come. Fill up here. Can I?
No issues. Easily.
He is taking photo.
I think it's better to switch it. Everyone having eyes.
This is the one, huh?
I don't know. Long one. I think this one.
Oh, you can.
No, this function is not available. I need to switch it.
No, switch off means.
This is the direct one. Yes, sir. Direct from here.
Have to switch off the LCD.
You take photos.
Your teachers do that.
Then the password.
I only have two hands. I'm not a Ashta Durga.
That's Durga now.
Whose phone is this? Oh, it's my only. Sorry, I forgot.
Then give it to me.
Yes. I'll not return these phones now. Sorry, huh?
For you, huh?
I'll not return. Huh? It's for me only. Okay. One by one. Huh? Okay, then ready, huh? Wait. Okay. Okay, sir. Ready? 1, 2, 3. One more. Okay. Ready. Okay, ready then. Okay. 1, 2, 3. One more. I think a few students they are wearing mask and just remove the mask. How you will identify it's me? You can identify nobody else.
It is easy to identify. They only over mask.
Okay, ready then. 1, 2, 3.
No freestyle.
Again, last time.
Last one.
Yes, last one. No, but because after this somebody will come here because I'll go there. No, I'll have the photos also. Okay, ready, huh? 1, 2, 3. Yes, one more. 1, 2, 3.
Then ask for freestyle.
Freestyle.
Jumping style. Okay, ready? Yeah, freestyle.
Boomerang.
Boomerang. Okay. Ready? Okay, ready? Yeah. Oh, mine no. Now it's mine. Mine only, yes. Okay, ready? No, not taking selfie, yours only. Now I'll take selfie. Oh, somebody's hiding, huh? Okay, ready?
You are ready.
Now somebody come and take the photograph, please.
Use back on.
Whose phone? No, I don't have my. It's all my phones now. Did I take phone from anyone? I didn't.
You have seen?
Huh? Yeah, it's video. Huh? Okay, thank you.
No hiding.
1, 2, 3. 1, 2, 3. Freestyle. Thanks.
Little time.
Okay.
Hello, come and tell the phone.
Phones? Whose phone? It's mine only. Mine, sir. Is it from? No, I was having 4 phones actually. Okay, whose phone is this? Yours, okay.
Please come and take your phone.
Hey, I have these 3 phones, please come and collect them. Oh, it's yours, sir. This one? One more I have. Anthony sir. Anthony sir. Group photo Vivekananda. Sri.
Oh. When you presented the winners, can you just project my name once again? Just want to take a picture from
Jamna is there, Anthony is there.
Yes, yes.
Sir, Prof, I have one claim form now.
because I need to send.
Need the photography for photo session now. So I'll fill the file of form. 500 ringgits is okay. Paisa, everything but paisa. No. Just sit and I'll take everything will be covered. That's the only reason I bought. Motorola. Not use there. No service center. Okay, ready, huh, everyone? Okay, yes, ready. 1, 2, 3, ready, huh? Back one, yeah, ready. Okay, 1, 2, 3. One more. 1, 2. Sir, please come, sir. Somebody call Anthony sir. Anthony sir. Come, please. Circle. Sit. Yes, everybody is happy, huh? Ready? Okay. 1, 2, 3. One more.
You should take using this now.
Many phones.
Can stop
Again,
already, I think.
I can share. I can